members, the respective members of the evaluation committee. And now uh, I give time and I invite them to kindly take over and conduct the proceedings. In this committee, sir, is it okay to reveal the names of the committee members?
Yes. Shall I tell it?
Committee members can be read out. No, it is not a secret matter.
Okay.
Okay. So the e-posters will be evaluated by committee, and this committee is convener of the committee is Dr. Nongren Khumba Singh Laikang Bam, who is the convener. He is associate professor and head Department of Plant Pathology, CAU Imphal. And Dr. Pranav Datta is member, associate professor, School of Crop Protection, CPGS Barapani, Meghalaya. And Dr. Lanje Wangdi, member, associate professor, Department of Botany, Sikkim Government College, Gangtok. So I request you to kindly take over and conduct the proceedings.
So thank you very much. Getting a privilege to be a main jury, so I really extend my warm thanks to all of you, especially Robin Gogoi, Susan, and whatever involved in this symposium. So my request is that all the candidates are present here. So shall we start it from serial number 1? Hello?
Yes.
Shall we start it from serial number 1?
Certainly, sir. You can read out the name and call the presenter.
So so please kindly represent the poster Bitsaha Barua. So, and also from my side, I request our members, Pranav Datta and Dr. Linde Wangdi. So please kindly be alert. Is my request to all of you. So first upon from my side, please arise any sort of question related to their poster related.
Sure, sir.
Okay, thank you.
So, Bidisha.
Yes, sir.
Yes. You started. So that is permitted only 5 minutes. So take your time. Please, you started.
Hello.
Yes, sir.
Yeah, I am in the webinar of IPS. Are you also? Have you joined? Okay. It is provided by Tia Au. In mail you will find Susan. Yeah. I called earlier to know and to have discussion with him. Okay. No problem.
So Bidisha, just proceed for your post-harvest disease of major fruits and their management strategies.
Hello?
Yes.
Am I audible?
Audible but not visible. Your this thing is Bidisha. Yeah. Your video is off.
Yes, sir.
Yeah. Yeah, you start representing, or you can take only 5 minutes. Okay, within that you have to finish.
Okay, sir. A very good morning to all. Myself, Vidisha Borwa, and my e-poster presentation topic is post-harvest disease in major fruits and their management strategies. So, as we all know, the disease which develop on the harvested parts of the plant, like seeds, fruits, and also on the vegetables, are the
Just
post-harvest disease.
Vidisha, just to hold on.
Yes,
Please
sir.
share your slide.
Yeah, your slide is not PPT.
Your
Okay, sir.
your
Yeah,
PPT.
poster presentation.
Yeah, please share your PPT.
Photos.
So, is it visible?
Just wait.
Yeah.
Yeah.
Yes.
Yes.
Yes. Yeah, you start explanation.
Um, the disease which develop on the harvested parts of the plants, like seeds, fruits, and also on the vegetables, are the post-harvest diseases. The harvested products may get infected on the way to storage or to market or even before their final consumptions. So basically, I take three major fruits, that is banana, grapes, mango, and papaya.
Yeah.
So as we all see here that the three major post-harvest diseases of banana are anthracnose, crown rot, and cigar and rot. And the grapes, for the grapes, there are four major post-harvest diseases, and they are anthracnose, or we can say bird eye spots, Brotres buns rot or grey molds of grape, blue molds rots, and rhizopus rot. For mango, there are several diseases like anthracnose, stem and rot, black rot, brown rot, etc. And for papaya, there are three diseases. They are anthracnose, stem and rot, rhizopus rot, etc. So their managements are as follows: pre-cooling method, natural fungicides, refrigerations. That's it. Thank you.
So it's over.
Yes, sir.
Okay. So please kindly advise our members. It is open to the juries, open to the house.
Okay, Vidisha, very good presentation.
Yes,
So I
sir.
have one query: whether you have
Thank you, sir.
isolated the organism?
No, sir. Actually, this is my dissertation of in MSc, and due to this COVID, I only collect the secondary data. It is basically on the review work.
Or it is their review work? Okay. So no isolation
Yes,
was
sir.
done during the course of your study, right?
No, sir.
Okay. Thank you.
Yes, sir.
So this from
Thank you,
my
sir.
side, I'm telling that as you have given the this thing topic post-harvest disease of major fruits and their management, it is too vast by
Yes, sir.
saying. So that should be specifically mentioned. Okay, the banana, major
Yes,
fruits
sir.
of banana, grape, mango, and pineapple. If your just topic is in your
Papaya.
dissertation, it will be very difficult.
Ye-
Everyone will arise the
Yes,
question.
sir.
Okay. So,
Okay,
so
sir.
Dr. Lindsay, please kindly from your side.
Okay, thank you, Vidisha. It was a good poster presentation. And I just wanted to know
you, ma'am.
why did you take this post-harvest diseases for your dissertation? Any specific?
So, ma'am, basically, yes, ma'am. Ma'am, sir, honestly speaking, it's given by my mentor, my guide.
Yes,
So I did it.
from your guide. Okay, okay, thank you.
So again, I am supplementing that as you have your guide has given it, why you are willing. So is there any I mean merit, demerit? So how you differentiate the merit and demerit of just taking dissertation? Tell me, do you have any idea?
Sir, can you
What I'm saying is that
sir,
as
can
your
you
guide
please
has
repeat?
given such topic, so you are willing, isn't it? So I think
Hmm.
your guide has convinced to
Yes, sir.
you. So why you are accepting as your guide has given such
So,
topic? Tell me.
sir, uh, the topic is quite interesting also,
Yeah,
and
yeah.
as
The
I am a
reason
student of
reason is just tell
botany,
me reason.
and so I want to know about the diseases of fruits. Uh,
Yeah,
We,
that's
we
fine. That is fine. Okay, I'll just
okay.
put up one small question. So, what is the causal organism for this whitish gray spot in grapes? That's it for, I think. Can you name any
grapes,
pathogen?
Brotres,
What?
causal organism of Brotres buns rod.
Whitish gray spot.
It's
What is the pathogen? What is the cause of this gray spot? Reason? Causal
gray
organism?
spot. It's Botrytis cancri.
Okay, thank you. I think her time is also over.
Yeah,
She
yeah.
started, so we can proceed with the next one.
Okay, that's fine. We can move to next presenter.
Thank you, sir.
Yes, thank you, thank you. So next one is uh, Bharasa Sharma. He or she is going to represent management of seed-borne fungi of French bean.
E. Narasimha will start.
Okay, sir.
Oh, you are ready?
Hmm.
Hello, Bharasa.
Yes, sir.
Yeah, just put on your video.
Okay, sir.
Okay.
Take your time.
So
Okay.
that's pretty
Sir,
Wish you all the
sir,
best.
sorry, sir,
Which
I
one?
cannot share
Pardon?
my screen.
You cannot present. Bharasa, what do you say?
Sir, actually, I cannot share my screen.
Because of what? Network.
I think no,
No,
Vidisha
no.
has to stop
Vidisha
sharing.
has to, yeah, Vidisha
Sir,
has to
please
stop
stop
her
sharing the
sharing.
screen. Okay.
Yeah.
But
Hello, sir.
yes. Good morning.
Good morning, sir.
Yeah.
Sir,
Please
is it
carry on.
possible?
Take your time.
Thank you, sir. Good morning to all of you. Myself, Barasa Sharma. Today, I am going to give you an online presentation. Hope you all like it. So, the topic of my presentation is management of seed-borne fungi of French bean with fungicidal treatment. So first come with introduction. Generally, seeds of French beans are infected during storage condition, which affect the germination percentage as well as root or shoot length. Seed treatments, mainly by applying fungicides, are the most effective means to control the seed-borne fungi. For this study, uh, so for this study, firstly, seeds of French beans were collected from different local farmers of Tezpur. Secondly, the surface microflora was isolated by agar plate method, and identification was made by preparing slides of fungi. Thirdly, 4 fungicides, namely Captain, Diethane M45, Jim 50, and SAF were selected for controlling the seed microflora by agar plate method. The germination percentage and root shoot length were recorded for several days. I was found that the seeds of French bean harbored altogether 7 fungal species, namely Aspergillus flavus, Aspergillus niger, Fusarium solani, mucor species, Rhizopus oryzae, Rhizopus stolonifer, and Penicillium species. The efficacy of the fungicides showed that at the 0.3% concentration of Diethane M45 and SAF almost completely eliminated the seed microflora of French bean, which is followed by Zim 15 and Captain. The untreated seeds showed more percentage of infested seeds. Seed microflora directly injure the seed, resulting in reducing percentage of germination. The untreated seeds of French bean showed 70% of germination percentage, where as Diethane M45 showed 96.67%. Sorry, sir. Sorry. (96.67%) are the improved germination of seed after fungicidal treatment is due to the elimination of fungi which are responsible for the reduction of seed germination. After fungicidal treatment, the growth of root and it is only because of some fungicides also acted as growth regulating hormones, and best results were found from Diethane M45 treated seeds. Although all the 4 fungicides tested were found to be quite effective in controlling the seed microflora of French bean, best results were obtained from Diethane M45 and treatment, as it eliminated almost all the fungi and improved the seed germination potential of French bean. So, at the end of this presentation, results of this study suggest that seeds treated with fungicides are good for planting. Diethane M45 is the best fungicide for the control of seed-borne fungi. Therefore, it is recommended that seeds of French bean should be treated with Diethane M45 prior to planting for a good harvest. That's all. Thank you very much.
Okay, now it's open to the house. Please, any queries?
Okay, Barsha Sharma, very nice
Yes, sir.
presentation. Thank you
Thank
for
you,
your
sir.
presentation.
Thank
So, you see, you have isolated the fungal flora from the seeds of French bean, right?
Yes, sir. Yes, sir.
So, can you please tell me how you avoided the growth of bacterial organism in your media or in your, say, agar plate method?
Bacteria. Sir, actually, bacteria.
It's okay, fine. So my next question is, you have mentioned some of the organism or the say pathogen up to species level. Then by what method you followed for identification of that organism up to species level? Can you please tell me?
Sir, it was just a morphological study. I made
But it is,
some fungal slides, and I identified a fungal pathogen.
but it is very difficult to identify the species level of that organism, especially say Penicillium or say Aspergillus. Up to genus level, it's okay. That's why I raised the question. It's okay. Thank you. Thank you for your presentation.
Thank you.
Okay, okay, from my side.
Okay, sir.
So, how you have done it? I mean, this thing, confirmation of the causal organism which is involved that particular disease. How you confirm it? You have
Sir, please repeat
isolated the
your question.
organism from the seed.
Hmm.
So, how you confirm that this is the causal organism of that particular seed? So, please tell me some few hints.
Sir, actually, when I put some fungicidal treatments hits, there is no pathogens, and without fungicidal treatment, which seeds were grown, that's I found those pathogens.
But you didn't do this thing, Cox procedure, general procedure. You didn't do that. For confirmation, at least you need it for isolation, and these are fungal isolated, which can cause the seed-borne disease. So you have to make out it. Okay. Anyway, just pass to
Yes, sir. I have,
Madam.
okay, Varsha, thank you very much.
Thank
So
you,
I have
ma'am.
a small question, because most of the isolation you have and identification you have done through morphological only, right?
Yes, ma'am.
So I think you must have isolated Aspergillus niger in the media Petri plates, right? So what is, I just wanted to know, how does this Aspergillus niger look into your Petri plates? Maybe it's 4-color or it's mycelium color. Have you?
Aspergillus niger is ma'am dark brown color. Actually, colony is dark brown color.
It's 4-color, yes?
Spore color, ma'am. There are conidia radiate and
Is it,
conidiophores are long.
yeah, is it dark in color? You have seen that in your culture? Black,
Yes, ma'am. Dark brown color actually.
black, brown. Okay, right. Thank you.
Thank you, ma'am.
So please, next candidate, sir. So please proceed. Lyro, so I could not pronounce it. So the serial number 3, in vitro compatibility of Trichoderma aspergillum with fungicide and its effect against important plant pathogen in Nagaland. Narola, she's present. She or he, I don't.
Yes, sir, I'm present.
Okay,
Sir,
okay.
am I
I'm not
audible?
wrong. She, okay. I could not. I can what to say. Tell that she. Okay. So please
Okay,
proceed
sir.
and take your time. Wish you all the best.
Okay, sir. Good morning, one and all. My name is Lalro Hlui. I'm from Sasert, Nagaland University. The topic I'm going to present is in vitro compatibility of Trichoderma aspergillum with
We can't see your
fungicides and its
poster
effect.
video.
Oh, sorry, sir.
Please kindly check it.
Okay, sir.
Saya diam tahu Roman ni.
Sir.
Yes.
Is it now?
No, yeah, it's about to come. Yeah. Open it. Open your watcher. Yeah, it's coming. Carry on.
Okay,
Make
sir.
it a bit
Okay.
enlarged. Okay. Make.
Is it
Yeah.
okay?
Yeah.
Sir,
Thank you.
my name is Rohlui. I'm from Nagaland University. The topic I'm going to present is in vitro compatibility of Trichoderma aspergillum with fungicides in effect against
So
important
network
plant
problem.
pathogens in Nagaland.
Your
Introduction.
voice is not coming nicely. So tell me some word. So clear or not? I want to confirm. Yeah,
Hello,
it's
sir.
coming.
Is it clear?
Yeah, it's
Okay.
correct. Please carry on.
Okay, sir. Trichoderma species are a ubiquitous fungus known to have an ability to be soil acid-borne plant pathogens. Materials and methods. To evaluate effect of Trichoderma aspergillum with important plant pathogens was done by dual culture technique given by Nice and Webster 1971. Investigation of compatibility of Trichoderma aspergillum with fungicides. The formula given by Vincent 1914 was used.
Sound. No, it's not coming properly. Your voice is not coming. It's breaking.
Coming properly now.
Yeah, not yet. Just make it clear.
Hello, sir. Is it clear?
Yeah.
Okay. I think it's because not that good. Sorry for that. I'll continue. Compatibility of
No, again, we cannot listen clearly your voice. So you are using this thing, headphone, microphone. You are not coming at all.
Oh my God, sir. Okay, sir.
You are making very
Sir,
close
hello?
to the microphone. Don't close very much to the microphone. Your, I mean, mouth. Okay.
Okay.
Yeah.
Sir, is it clear now?
Yeah.
Okay. Compatibility of Trichoderma aspergillum against different fungicides. I've tested 5 systemic fungicides, and among these, azoxystrobin was the only systemic fungicide which was comparatively compatible, while carbendazim, thiophenate methyl, Kitazine and propiconazole were all incompatible. And for contact fungicides, mancozeb, copper oxychloride, chlorothalonil at lower concentration, that is 0.1% and 0.2%, they were comparatively compatible. But progressive increase in the concentration leads to incompatibility. And for dual culture technique, antagonistic activity of Trichoderma aspergillum against important plant pathogens. 4 common pathogens were collected from the department: Sclerotium rossi, Rhizoctonia solani, Fusarium oxysporum, former species Lycopersicii, Alternaria solani. And among these, Alternaria solani showed maximum inhibition, 77.76%, followed by Fusarium, 73.26%, Sclerotium rossi, 62.93%, and Rhizoctonia solani, 57.7%. And the pictures can be seen here below. For future aspect, combination fungicide with antagonists should be further studied to provide information on the dose and time of application. And this can be incorporated in integrated disease management method for more effective and sustainable plant disease management. Thank you. Hello.
So it's open to the house. Any queries, any questions?
Yeah, thank you. Thank you for your very good presentation. And it is actually need of the hour to see the combination of fungicide as well as some biocontrol agent. But here I have one query. Why you have chosen Trichoderma spirillum? As because we know a huge number of Trichoderma species are there, say like Trichoderma harzianum, Trichoderma viridi, Trichoderma hamatum, longibricatum, pseudocorengi, huge number. But why you have selected this Trichoderma spirillum?
Sir, I have selected Trichoderma aspirillum for a new future prospect because lots of studies have been already done for Trichoderma harzianum, Trichoderma viridi, etc. So I've selected this Trichoderma aspirillum.
So if I say that means inhibitory ability or disease suppressing ability of other species of Trichoderma are more as compared to the isolate you have selected for your study, say Trichoderma spirillum. In that
Yes, sir.
case, if it is a new or whether as it is a new, should we take it or we should concentrate on the other potential biocontrol agent like say Harziana viridi or like that?
Sir, for me, I think we should also take it because we can't neglect all these essential biocontrol agents, and then we can study more about their compatibility effects with with this fungicide again.
So how many doses you have selected or you have tested to test your compatibility? Say suppose you can take
Sir,
one example, thiophenate methyl or say carbendazim. So how many doses you have considered for your study?
sir, I've considered three doses of concentration: 0.1%, 0.2%, and 0.3%.
What was the base of selection of this concentration?
Sir, I've searched some literature review, and then most of them were most of them I have seen it in lower doses of concentration.
Actually, we
They have
used to
studied in lower doses of
do,
concentration.
yeah, you used to do the study like that.
Yes, sir.
Lethal dose, sublethal dose, and higher dose. Okay, that is
Mm.
recommended dose. Then one lower concentration and another is higher concentration. It's okay. Now my
Yes.
last question is that which method you used to study your compatibility? I could able to understand to study the efficacy against the your pathogen. You used dual culture method, but for
Yes, sir.
compatibility test, which method you have used?
Sir, I'm using poison food technique.
Okay, okay. Thank you for your nice
So,
presentation.
only in supplement of this, your the specific trichoderma is being used for your study. So, it is native or you have selected from, I mean, outside your state. Is native one or from other state you procure it?
Sir, it's from Nagaland, and then I obtained it from the department.
Okay, okay, it's very good, very good. So, madam, please.
Okay. I wanted to know because most of my concern was already put up by Dr. Datta. So my only concern is that what is that? That means this you are using the fungicide along with this trichoderma. So that
Yes.
means this fungicide is not affecting the potential of the trichoderma, biocontrol potential of trichoderma.
Ma'am, some are affecting and some are not affecting.
Affecting in the good sense, or that means it is affecting in the bad sense, that means it is controlling the potential, biocontrol potential of fungicides, or when you combine these two together, it will give a better result?
Yes, ma'am. Yes, ma'am. I assume that one because fungicide. For example, mancozeb. Uh, for example, mancozeb. This Trichoderma aspirillum, they degrade the fungicide and utilize their product carbon for its growth and development. So I'm assuming that it will be more effective.
Effective when you combine this
Yes,
along with the fungicide.
yes, ma'am.
That means this fungicides cannot kill this trichoderma, right?
Ma'am, in higher dose of concentration,
Okay.
it can be killed.
So that may be from the recommended dose.
Yes, sir.
No. Half of the recommended dose, because she already mentioned 3 different doses she has used. So probably at lower dose she could able to get the compatibility, and in the field condition there may be some synergistic effect, because lower dose will partially make weak the pathogen, and a weaker pathogen is easily attacked by a biocontrol agent. Probably that is the probably the hypothesis. Still, it's okay. Thank you.
Okay.
Thank you,
Very
sir.
good. Okay. So thank you very much. So next contestant is Hemant Hazong. Please kindly carry on. So your poster presentation in vitro evaluation of certain bioagents and fungicides against Rhizoctonia sonali. So Hemani, hello. Yeah, you are present, isn't it? Hello, Hemani. Hello.
Hello, sir.
Yes.
Yes, sir.
Yeah, please carry on. Show your photo. Yeah.
Yes, sir.
Yeah. So little bit magnify again. Yeah.
Yes, sir.
Yeah. Please
Uh,
carry on. Take your own time.
Thank you, sir, for the time. Good morning to one and all. My name is Simani Hajong from Nagaland University, Sasar, and the topic for my poster presentation is in vitro evaluation of certain bioagents and fungicides against Rhizoctonia solani. Introduction. Disease caused by Rhizoctonia solani occurs every year, everywhere, and causes yield and quality losses on several important crops. Materials and method. To evaluate the efficacy of bioagents against Rhizoctonia solani was done by dual culture technique after Dennis and Webster 1971. And for calculating the percent inhibition, the formula used was control minus treatment by control into 100. And for to evaluate the efficacy of fungicides against Rhizoctonia solani was done by poison food technique after Nene and Thaplial 1982. And the formula for calculating percent inhibition is control minus treatment by control into 100. The experiment was conducted in a completely randomized block design, and each treatment was replicated 3 times. Table 1. In vitro evaluation of biogens on percent inhibition against Rhizoctonia solani. From this table, it can be seen that
Don't go through all. It's just which one is the most effective and which one is not giving low effective. Only that you mentioned. Okay.
Okay, sir. Okay, sir. From this table, it can be seen that Trichoderma herzianum 14 gave maximum mean percent inhibition with 58.96%, and the lowest inhibition percent was observed in Trichoderma virens with 30.98%. And below the table, these are the pictures showing the in vitro evaluation of biogens against Rhizoctonia solani. Table 2. In vitro evaluation of fungicides on percent inhibition against Rhizoctonia solani. From this table, it can be seen that mancozeb at the rate of 0.25% and copper oxychloride at the rate of 0.3% showed showed 100% inhibition, and the least inhibition was observed in azoxystrobin at the rate of 0.1% with 39.25%. And below the table, these are the picture showing the in vitro evaluation of fungicides against Rhizoctonia solanae. Result and discussion. In dual culture technique, Trichoderma harzianum 14 showed the highest mean percent inhibition against Rhizoctonia solanae with 58.96%, followed by Trichoderma harzianum, Trichoderma spirillum, and Trichoderma virens with 59.97%, 40.95%, and 30.98% respectively. And in poison food technique, Mancozeb and copper oxychloride showed 100% inhibition when tested against Rhizoctonia solanae, and which was followed by chlorothenolil and azoxystrobin with 68.69% and 39.25% respectively. Thank you.
So it's open to the house for any questions.
Thank you, Himani Hajam, for your very good presentation
Thank you,
on
sir.
in vitro evaluation of bioagent and fungicides against a dreaded pathogen, that is Rhizoctonia solanae. But
Yes, sir.
while presenting, you have mentioned your experiment, probably as I understand, your experiment was on in vitro evaluation, right?
Yes, sir.
Which statistical design you have used?
Sir, completely randomized design.
Oh, that is CRD,
CRD.
right?
Yes,
Okay.
sir.
Can you tell me what is the difference between Trichoderma virens and Trichoderma, say, another species you used, Harziera, right?
Yes, sir.
Can you please tell me?
So actually, I haven't studied their morphological characters.
Oh, you have not observed under microscope how Trichoderma look like, so beautiful structure.
Yes, sir.
You never observe under microscope?
Sir, I have observed some Trichoderma species, but then I haven't compared them.
Okay. What is the main mechanism of this Trichoderma like biocontrol agent to suppress the pathogen like Rhizoctonia solanae? What are the different type of mechanism? Why
Sir,
they are
it's
suppressing?
sir, because of the mechanism of antibiosis, competition, and mycoparasitism.
Whether you have observed under microscope that mycoparasitism or say coiling or say lysis?
No, sir.
It's okay. Thank you. Thank you for your presentation.
Thank you, sir.
So, ma'am, oh, she has gone out. So anyway, Himani.
Yes, sir.
So you have done it everything, isn't it? So
yes, sir.
How is the difference between fungi and trichoderma? Just tell me.
Fungi.
Am I
Sir,
wrong raising such question?
no, sir. Sir, Trichoderma also belongs to, is one kind of fungi only.
Yeah. So
Yes,
I
sir.
want to know whether you have clearly got known to you. So I'm asking. Okay.
Yes, sir.
So wish all the best. So,
Thank you, sir.
ma'am, she has gone audit. So okay. Thank you very much.
Thank you, sir.
Yeah. So now comes to Varsha Devi. Miss Varsha Devi.
Hello,
She's
sir.
present. Yeah, very good. So she is going to represent about the post-harvest disease of banana and their management, a case study in Greater Tezpur, Assam. Let's proceed. Carry on your own time. Okay.
Good morning, everyone.
Yeah, make it enlarge. Okay, magnify it.
Good morning, everyone. Today, my topic is post-harvest disease of banana and their management, a case study in Greater Tezpur. Post-harvest disease may start before or after harvest. Once it is stored, the infection of the disease develops on the fruits and vegetables. As we know, banana is rich source of carbohydrate and vitamins, and also good source of potassium, phosphorus, calcium, and magnesium. At first, the disease samples from different market of test pool were collected. Then isolations were carried out from the crown portion, roted pulp, ready spot, direct tip, and rot. Uh, then washed thoroughly with tap water. Disinfected tissue were isolated in PDS slant. After isolation, identification are done on the basis of morphological of somatic and reproductive structures by preparing slides. Then pathogenicity test was done by pricking method. But for controlling disease, two methods are used. Dipping method for crown rot, cigar and rot, and hot water treatment is effective for anthracnose disease of banana. For dipping method, all the banana are dipped in fungicide, carbendazim, mancozeb, and captain. Some symptoms are observed in present investigations are described below. Musa sempiantum vervim. Symptoms observed in infected Musa sempiantum were softening and blackening of tissues at the crown surface. Musa nana var jahazi. Infection appears as numerous small, circular, and brown to dark brown spots. Musa nana var sagar. The infection appear with localized darkening and wrinkling of the skin, originate at the perianth and spread slowly backward along the finger. Different fungi were successfully isolated from different banana fruit rot, included crown portion, ready spot. On the basis of cultural and morphological character of isolate were identified as Fusarium species, Coledotrichum species, and Verticillium. For pathogenicity test, healthy banana were inoculated with 7 old culture of Fusarium species, and result observed that the symptoms usually appeared on the Musa sempiantum is same as the previous symptoms. Thus, the cause postulate was established and a disease was found to be crown rot of banana and caused by Fusarium. Likewise, same procedure were followed by Coledotrichum and found that the disease is anthracnose of banana and is caused by Coledotrichum. Similarly, same procedure for Verticillium and found that cigar and rot caused by Verticillium. When the fruits were incubated at room temperature treatment with mancozeb did not give a satisfactory result, protection of fruit as regard to carbendazim and captain. Dipping in carbendazim solution allowed keeping more than 95% of fruits free of disease for 7 days. Here I have shown some pictures how we can establish cause postulate of these 3 types of disease of crown rot of banana. Crown rot disease caused by Fusarium, then anthracnose disease caused by Coledotrichum, and cigar end rot disease caused by Verticillium. Here I have also shown some pictures of controlling these three type of disease by using different fungicide like carbendazim, captain, and mancozeb. Now finally come to the conclusion. Many pre-harvest factors directly and indirectly influence the development of post-harvest disease. Traditionally, fungicide has played a central role in post-harvest disease control. In the present study, fungicides are used in very low concentration so that the environment does not get affected. And with this, I concluded my topic. Thank you. Thank you, sir. Thank you, sir.
Yes, thank you, Varsha.
Hello, ma'am.
Yeah, can you hear me? Yes, it was a good presentation. And I just wanted to know, to sum up, what is, because it is, your session is in full, what is the thing out of your work, your future technologies, yeah, some kind of innovation? Just sum up your entire work in 1 or 2 lines.
Ma'am, I cannot hear your question, ma'am. Ma'am, can
Yes,
you
your,
repeat
your
your question,
innovation,
please?
what is the outcome of your work?
Ma'am? Hello, ma'am?
Yes,
Hello?
what is the outcome of your work, or what is the technology? What?
Ma'am, mostly
Sorry,
the,
sorry. Okay,
hello?
go on. Yes, yes, go on.
Hello? Hello, ma'am, mostly banana fruits,
Yes, yes, we can hear you. I can.
mostly the banana are infected when stored in is infected by various fungi. So I identify the disease and how we can manage the disease, and you can, and we can use for future purpose. By managing, we can use for future purpose.
Okay, okay, thank you.
Okay, Barsha.
Yes, sir.
So you have you worked on 3 different type of diseases, post-harvest
So yes.
diseases.
So
So
yeah.
according to you, which one is more devastating, and because of which the shelf life of banana is drastically reduced after, say, harvest?
Sir, all the three diseases are basically drastically means damage the fruits, but anthracnose is the most disease that drastically damage the whole fruit. So I have shown the pictures of these diseases.
So based on your work, can you suggest 1 or 2 good management practices to avoid these 3 diseases on which you have studied?
Sir, I have selected three fungicide, generally carbendazim, then captain and mancozeb. And with this, I have found captain and carbendazim give a best result in regard to mancozeb. So I sir recommended carbendazim and captain. By using these 2 types of fungicide, we can manage properly.
So can you please tell me, after, say, spraying of this type of fungicide, after how many days we can consume the harvested product?
Sir, I have used a dipping method for this.
No. My question is, what should be the waiting period?
So for 7 days, we can get the result. Sir, for
No.
experiment,
Say suppose you have suggested a farmer to spray this type of fungicide, okay,
yes, sir.
carbendazim or semencozeb or captan. Now my question is that, so after how many days, as a consumer, I can consume your treated uh, banana?
Sir, for the, sir, in doing this experiment, sir, for 7 days, there is no diseases for the banana.
Really, for most of the fungicide, the waiting period should
I
be at
think
least
period
12
is
to
there. So he,
20. Yes,
she's not
yes.
getting.
It's okay. It's okay.
Uh,
It's okay. Thank you. Thank you for your nice presentation.
okay, thank you, thank you.
Thank you, sir.
So shall I call out MA Kam Wamat? Nung Tadu. So please kindly, so MA Kam, what is there?
Yes, sir.
Yeah. You from which state?
Meghalaya, sir.
Oh, Meghalaya. So because of that, name is very difficult for me to pronounce. Okay. Anyway,
Yes, sir.
it is our pleasure to be here. So just take your time. You are going to represent about the evaluation of fungicide compatibility. What is potential biocontrol agent for management of chili and anthracnose? So please take your time.
Yes, sir. visible?
Yes, it is coming.
Okay. Hello, very good afternoon to you all. Our respected Chairman, Sir, and all the participants present here. The topic that I would like to share with you is this poster presentation is evaluation of fungicide compatible with potential biocontrol agent for management of chili anthracnose, which is caused by Polytrichum capsaicin. As we all know, chili is one of the most important commercial crop of India, and not only in India, but the very important crop. And mostly we are used in every kitchen use, and it treats. Hello.
Yeah, we can hear you.
Okay, sir. I'll begin it again from the introduction. Chili is one of the most important commercial crop in India, and not only in India but in the whole worldwide. It's a very important crop, and but due to the several diseases, it used to cause heavy yield loss. And one of the most major constraint in chili production is due to the anthracnose or fruit rot, which is caused by a pathogen called Colostotrichum capsaicin. And there are several management were approach and various combination of method that leads to integrated disease management in order to control the disease. However, after all these methods, still this fruit rot continue to be the major constraint in the production of chili. And therefore, in the present experiment, I tried to focus on identification of the fungicide which is compatible with the biocontrol agent against the pathogen, which is a Colostotrichum capsaicin, develop an effective management strategy. And first, what it is, I tried to collect the sample of chili, which show the characteristic symptom of the disease, and I tried to isolate the pathogen. After 2-3 days of incubation period, after the mycelium is full in the plate, I tried to take a portion of it and put it in a slide and try to observe under the microscope in order to consider the this pathogen is causing causing diseases is the right one or not, because there are so many Colostotrichum, and this species we can identify.
Please continue.
So
Okay.
I was very sorry. The current has gone and now again came back. I was trying to. So she has completed.
No, sir. I'm still because of my network
Okay,
issue.
carry on.
Okay, sir. And then I try to select 5 fungicide against this pathogen. I try to evaluate it. The name are hexaconazole, propiconazole, carbendazim, copper oxychloride, and azoxystrobin. And all these fungicide I use it according to the recommended dose. And then for the antagonist, I try to collect 5 antagonists: Trichoderma arzianum, Trichoderma virens, Trichoderma aspergillum, Trichoderma species 1, Trichoderma species 2. These 5 Trichoderma, after collecting it, I try to maintain it its pure culture, and then for the further studies, and I try to subculture it after a 7 days interval in order to maintain the purity of the culture, and I store it in 4 degree Celsius in refrigerator. So after the method, the result and observation I got from this is the fungus confirmed as a Colotrichum capsicum based on the homological characteristic. This colony of this particular pathogen is white to grayish and olive green color after its full growth, and this is the CT on the 40x, and this is the conidia on the 40x, and this is the conidia 4 on the 10x. And then from the poison food technique, among all the 5 fungicides evaluated, carbendazim, propiconazole, hexaconazole, and azoxystrobin, which I use in at the rate of 0.1% against this pathogen, was found to be more effective with 100% inhibition. And copper oxychloride was recorded as the least percent inhibition, which is 76.40%. And the percent inhibition was calculated over the control. And then for the dual culture result, among all biocontrol tested, Trichoderma had all this Trichoderma species were completely heavy of this pathogen, but then the most effective with 77.39% inhibition is Trichoderma harzianum. So after getting the result from the this poison food technique and the dual culture, I came to know that whether this most effective potential biocontrol agent is compatible or not if we together with fungicide. So after that, I try to test the compatibility test. So the most effective fungicide, 4 fungicide are carbendazim, hexaconazole, propiconazole, and azoxystrobin. These 4 fungicide are tested with the most potential biocontrol agent is Trichoderma harzianum. So after tested compatibility test, I found out that the person inhibition over the control. Fungicide, the 3 are 100% inhibition, which means it's not compatible to be together to treat it together. And compared to azoxystrobin, it was completely least percent inhibition, 3.62%, which means that is the most compatible fungicide. So what I want to conclude from this my experiment is true rot of chili is very serious disease in all over India and mostly in northern eastern India, and then we should approach more management in order to control disease. And I will we better for further evaluation under in vivo condition also can be done in order to confirm their efficacy, their compatibility. In vivo it will be the same or not, and it could be included as component for the integrated disease management module for autonomous disease of chili. Thank you, sir.
So it's open to the house for any questions, please.
Yeah, I'll go. Can I ask one question? Can you hear me? No. Hello?
Nongdu, can you hear?
Hello?
What's the one to him?
Can you hear me, sir?
Yeah, your voice is clear,
Yeah, yeah,
but
I can hear, but
Emka, she
whether
should
she has left.
no, she should unmute herself. Emka, please unmute.
Oh,
Yeah.
yes, sir. Yes, sir.
Yeah.
Okay, can I
Okay, madam.
go first?
Yeah.
I'll just a small query. In your presentation, you have shown the conitophore of Colitotrichum capsaicei. No, in your slide, it was there.
Yes, ma'am.
So in that picture, are you sure is that a conitophore or any other structure? Because it is in 10x, right? 10x. So are you sure that it is conitophore of not any other structure?
I'm
Like
sure it's
Aprisoria, it might be Aprisoria. It looks like Aprisoria to me, not conitophore.
But yeah, means
so you can just find out. Yeah.
The
Afterwards, you can just go back to your lab and just see whether it's actually a conitophore or Aprisoria. Okay. Because Colitotrichum conitophore, I'm not so sure. So I just have some confusion. You can just confirm it, right?
Yeah,
Emeka.
And
ma'am.
Okay, thank you.
Emeka.
Yes, sir.
Only one question from my side. You have shown 10 eggs, 30 eggs, 40 eggs. You have taken the, captured the structure of Conidea and others. What do you mean 10 eggs or 40 eggs? Just give me hints to me, okay? I'm a layman. So you presume as a layman, what is 10 eggs and what is 40 eggs, 60 eggs, 100 eggs? What do you mean?
This is the lancer. In like when. Hello.
Yes. Tell me. No, don't take much time. If you're having doubts, so I'll clear it. So 10x means what happened? It can magnify 10 times. Okay. 40x
Yes,
means
sir.
40 times. So in the IPS, that is in IPS, if it is 10x and below is 10x, then okay, that means 10 into 10. How much? 100 times, isn't it? What, 1,000 times or what? 10 into 10, how much?
Hello.
Can you hear? Emeka.
My phone. Hello.
Emeka. You got
Yes,
the
sir.
idea? You got the idea?
Hello, sir.
Yes.
Hello.
Yes, I can hear you.
Yes, sir. Okay.
So you
Yes,
got the
sir.
idea right now.
Regarding what, sir?
10x, 40x, you have to know it. This is the magnification of the particular. Okay, you are observing under the microscope. 40x
Yes, sir.
means
It's the
40 times.
it's the magnification of the scope.
Yeah. So anyway, so what is the disease symptom of what you said it? The causal organism? Colitotrichum, isn't it? Yeah, just tell me the
Yes, sir.
symptoms of a particular pathogen which involves causing the disease. You just tell me the symptoms which is
It's
happen.
necrotic lesion is formed around the part of the fruits or in the leaf part. And especially in the wet portion of the chili, most of the time the lesion is very visible, and in the necrotic black circular sunken type is
Yeah. when it is happened to the fruit, what the main peculiar symptom? Just tell me.
In the fruit, it's some good type, and even the sir
Got it,
answer will
got it.
like
Yeah, I got it. I got it.
fruiting
You
body.
have the idea. Okay, so Dr. Pranab, please kindly.
Yeah. So, Emeka, very good presentation and very good work you have done. So, I have a very simple
Thank you,
query.
sir.
In your in vitro studies, after how many days
sir.
you took the observation to calculate the mycelial growth inhibition percentage? After how many days of your inoculation? Yeah.
That is, actually, sir, I used to take 24 hours interval, and then when the full growth.
Hello. Yes. Tell me. Hello. Emeka. Okay, Chairman sir, we can
Yes,
go
yes.
to next, because she is
Yeah. So thank you very
network
much,
problem.
Dr. Emeka. So
Hello.
in short, thank you very much. So next contestant is what is it? Tayenla. Tayenla, please kindly proceed. He or she is going to present about the effect of different substrate pre-treatment method on the growth and yield of what? Ice Almarius. Please proceed it.
Yes,
Take your own time.
uh,
Hello.
Good morning, Chairman, sir. Yes, sir.
Yeah.
Sir, am I audible? Good
Yeah,
morning,
audible,
Chairman, sir.
but not visible. Yeah, just
Sir, while we are using
unmute your video.
sir, is it visible, sir?
Yeah,
My
it's coming
post,
up.
sir.
Yeah, it's nice. Yeah, just magnify it. Yeah.
Good morning,
Carry on.
everyone. Respected Chairman, sir.
So, I mean,
all
don't
the
make it closer your mouth to the
participants. My name
microphone.
is Tiaila, and I'll be presenting
Hello.
on the effect.
Please don't make it closer to your mouth near the microphone. Little bit lengthy. I mean, maintain the gap, and you just deliver it.
And
Yeah. Carry on. Hello. Network problem. Hello. Can you hear me? Hello. Are you using headphone? Hello. Tayenla. Sir Pranab ji, what shall we do? What shall we do?
We can proceed for the next one, because she is having network problem probably.
Okay, so Tayenla, please wait. I mean, you will be provided when the network is available within this I mean short period. Okay. Okay. So now the next candidate is Supriya Sharma. So please kindly proceed. So her presentation will be study on disease of economically important medicinal plants of Jorhat district, Assam. Please. Supriya. Hello. Hello. You are present here or not? Supriya.
No, she is absent.
She is absent. Okay.
Yeah.
Then, so next one is Resmi. Resmi, you are present. Resmi.
Sir, am I audible?
Yeah. So your volume, just make it increase. You are present here, isn't it, Resmi?
Yes, I'm present. Sir.
Yeah, your voice is very small. We are listening. What to say? You are speaking from somewhat far away.
Sir, can you hear me now?
Yeah. So you just make out. Please carry on. So you
Okay.
are going to, present about the study on post-harvest disease of Rabi vegetable in Jorhat district of Assam. Please proceed.
Yes, sir. Yes. Okay. Good morning to all of you. Today
Yeah,
the
now
e-poster
little bit reduce
presentation.
your voice. So please kindly increase it.
Okay, sir, can you hear me now?
Yes.
Yes. Okay. Good
Now,
morning to all of you.
no, it is no. Your voice is very reducing. So increase.
Okay, sir. Am I audible now?
Yeah.
My voice is okay.
Please proceed.
Okay, sir. Can I start now?
Yes.
Okay.
Yeah, magnify
Good
your.
morning to everyone.
Yeah.
Good morning to everyone. And the e-poster presentation, the topic is seven post-harvest diseases of
No,
rabi vegetables
your
in
mouse
Jorhat
should not
district
be
of
closer to the microphone. Little bit gap you have to maintain it. Otherwise, the echo comes. Okay.
Okay, sir.
Yeah, you start. It's also having problem.
Yes, she is having
Minerals
problem.
and
Yeah, please carry on.
yes. As we know that vegetables are important source of dietary fibers, minerals, antioxidants, and vitamins, there is also losses in vegetables, which is caused by some post-harvest diseases which may occur from harvest to consumption at any time during post-harvest handling. This may be fungal pathogen or bacterial pathogen. We have to identify. As for example, some of the post-harvest diseases in vegetables may cause by Penicillium, Aspergillus, Jotyphus, botrytis, etc. The main objective of my project is to survey of local markets for collection of diseases vegetables and then to isolate, identify, purify and maintenance of fungal and bacterial isolates and to test the pathogenicity of pathogens in the vegetables. Here comes the methods and methods. For this, I have used two media, the PDF media and NM media, and then I've sterilized it and then I've collected the samples from various markets. Were in the pathogen and used for PDF media and for I've used NM media. Then I've tested the pathology postulate of we know that tomato is the culture where we can see that the colonies are fast growing in tomato and cottony and it's but whitish short and aerial high flat brown. Okay, and then there's a microscopic observation which is my Captured branch light brown. From this, it is confirmed that it is alternary ST. Next is the cauliflower. Here the conidiophore are darkly pigmented, septate, and showy-like branching. It is identified as cladosperm ST. Samples to this project, samples from this at become
Resmi, your voice is not clear. Resmi,
can you hear now?
your voice is not clear. You make it finer.
Sir, am I audible now?
Yeah, audible. Yeah, don't close your mouth. I mean, maintain the gap in between this mouth and microphone. Okay.
Okay.
A bit
Sir,
louder.
I'm
You
not
speak
using
it.
Okay.
Yeah,
And
carry
here's
on.
the
Carry on.
yes, okay. And here's the capsicum. I've used in this identification. The identification of capsicum is bacteria and it is gram negative, which is confirmed as Arvinia ST. And then I've taken the P where the hyphae and spores are white and appear colorless. And it is confirmed as deuterium ST. Here is the table which I've taken the samples, and these are tomato, cauliflower, capsicum, carrot, pea, chili, bean, and brinjal. Here the name of the pathogen of tomato is Alternia ST, and the disease is early blight of tomato. And for cauliflower, it is Cladosperm species, which is and the name of the disease is Cladosperm rot of cauliflower. And for capsicum, I've got Arvinia ST, which is Pexel rot of capsicum. And for carrot, it is Jotrica species, which is store rot of carrot. And for pea, it is Yersinia ST, which is white mold of pea. For chili, it was Botrytis ST, which was gray mold of chili. And then bean, which is Yersinia ST, which the disease is gray mold of bean. And for brinjal, the pathogen is Phomopsis species, and the disease is Phomopsis fruit rot of brinjal. And here's the conclusion. We can see that for the further study of various post-harvest diseases, their management strategy will help to reduce the losses after harvest. Post-harvest diseases of fruits and vegetables limit the period of storage, compromise marketing, and consume adaptability and disease rate losses. And then as a result, food and nutritional security will also be improved to a great extent. Thank you, sir. Hello.
Yours is over. Hello. Resmi.
Yeah, she has finished her presentation.
Yeah, but your speaker also not working when I speak. Your speaker also not working when you speak from there. Your microphone also not working properly. So that is due to the network. I don't know. Anyway, just it's open to the house. Any queries, please, madam.
Okay, Chairman sir, can I ask?
Yes.
Okay, Rashmi, very good presentation, good work.
Thank you, sir.
But you see, your work is a little bit routine work. Can you tell me what is the new in your work? Because almost all this information are available. Many people has done lot of work in this type of thing. So what is new in your work?
Sir, this is the dissertation work of MSc, which was done this year.
Okay. You could able to isolate numbers of organism. Now, can you tell me how Phomopsis look like under microscope?
Yes, sir.
Hello.
Yes, sir.
How Phomopsis look like under microscope?
Sir,
And what type of
cotton
conidia
is
they produce? Yes.
conidia observed as sub-cylindrical and high length.
For Phomopsis?
Yes, sir.
Are you sure? Because you please, my suggestion is to please see the organism Phomopsis and its structure under microscope. And it's okay. Thank you. Thank you for your presentation.
Okay, thank you. So shall we proceed for
Yes,
other?
sir.
Yes, yes, yes.
So Miss N. Shurmina Devi, are you there? Hello? And Shurmina Devi.
Yes, sir, I'm here.
You are doing high. It seems like it's after a long time, after a long gap, you are giving your response. Anyways, just proceed. You have to present exposure of important medicinal and aromatic plants, diseases of plant diseases of Arunachal Pradesh. Please proceed.
Yes. Good morning, everyone, and thank you very much for giving an opportunity
A bit
to
louder. You just
share
speak.
our initial.
And
Good
your
morning,
video is
everyone,
not
and thank
coming.
you so much. Sir, I have already shared my screen, sir.
Yeah, but
I've already
it's coming
shared
out,
my
coming out.
screen. Okay, good morning, everyone, and thank you very much for giving an opportunity to share our initial work on identification of the some of the important diseases of medicinal and aromatic plants in the East Siang district of Arunachal Pradesh. As we all know that Arunachal Pradesh is a small state, but it is the largest hill state of northeastern regions. And I must say that we are blessed to have 5 different agroclimatic zones in Arunachal Pradesh. And it is we also it is also one of the highest rainfall recipients with 3,500 mm in a year for a period of 8 to
Hello. Hello. So can you hear us? Hello. Samina. Samina, you're there.
Yes, sir, I can hear you.
Yeah,
Hello?
but we cannot hear. Now though I can hear. So please carry on. Please carry on.
Okay, sir. Good morning, everyone, and thank you so much for giving an opportunity to share our initial work on the exposure of a important medicine and aromatic plant in the East Siang district of Arunachal Pradesh. And as we all know that Arunachal Pradesh is a small state, but it is the one of the largest hill state of Northeast Himalayan region. And I must say that we are blessed to have a 5 agroclimatic zone in Arunachal Pradesh, and it is also one
you
of
are
the highest
delivering the
rainfall.
lecture. So you show your poster also, please. It's not coming here.
Yes,
Not
sir.
coming up.
I will continue, sir. I will continue.
Yeah.
So as we have a different 5 agroclimatic zones, and the soil and the climatic condition offer an ideal condition of a production of wide variety of vegetables and fruits, and we can say the orchids, and then the medicine and aromatic plant also. People are more focused in important disease of fruits and vegetables, and I think the research has done also mostly on the fruits and vegetable, but we ignore the medicinal and the aromatic plants, and as well as some of the ornamental plants. And these are some of the important medicinal aromatic plants that were commonly grown in northeastern regions. And here during this
So,
pandemic
Dr. Pranab,
period,
Dr.
we
is
have
not
found that.
coming her poster.
Sharmina, please,
Yes, sir.
yeah, please share your poster because your poster is not visible for us. Okay. Please
Okay,
share
sir.
your
I'll
poster.
share it again. Please give me a second, sir. Is it visible, sir, now?
Just wait. It will take some time.
Yeah, it's coming. It's coming.
Yes, yes. Now you can proceed. You give in a slideshow. You please click slideshow in your laptop.
Yeah, yes, sir. So, sir, see, these are some of the important map that commonly cultivated in the northeastern region. And during this pandemic period, we have found that people are more
Sharmina,
focused
please
in the
go to the
medicine
next
and,
slide. You are still in the first slide. Please go to the next slide. Yes. Now you can present.
So in this table, I want to highlight that see here some of the important diseases, some of the important medicinal and aromatic plants.
Again, gone.
Yeah.
Oops.
So the network is too bad out. Can I present other slides? It's
Yeah, you
fluctuating, so it's coming and going out here.
Yeah, you have to go to a place where network is you can get a good network. Okay.
It's coming and going. It's coming and going, sir. Normally,
Yeah,
it's difficult here.
please finish up. Please finish up. Now coming.
Yeah.
Okay, sir. Okay, sir. So here today is some of the important. I highlighted some of the important medicinal and aromatic plants here. And then during this pandemic period, people are more focused in the medicine plant to boost their immune system and all. See here, the black gingers also, the people are demand in the market also, it's very high in this pandemic period. And here, see the chameleon plant, which is
So now network has gone again.
Yes,
Sir,
yes.
what shall we do?
We should give her some time so that she can get a good place where network is not.
I mean, shall we proceed another one? Let her
Yes.
wait for some time.
Yeah. Serial number 11.
Sir.
Yes. After Shaminaji.
No, this is Lanzi.
Oh.
Her presentation, she has not prepared in that poster format, no?
Yes. That
She
thing
has
I have
made
also
in
noted.
because it's a poster compilation.
And what shall we do, madam?
I think once she joins, we can just ask her to conclusion, main gist of her presentation.
Yeah, shall we proceed another one before?
Yes, next.
I think, next, we should go for next.
Yeah.
Yeah.
Network is
So
the
now,
problem.
next candidate is
Chakruno.
Pejamguli. So, Pejamguli, you're there?
Yes, sir.
I'm sorry, I have spoken out very loudly, okay? Wish you all the best. You are going to present about the estimation of avoidable yield loss due to
Sir, I'm here, sir.
Pardon?
Sir, just okay, sir.
Okay. Please proceed. Take your time.
Okay, sir. I'll share my screen, sir.
Okay.
So is my screen visible, sir?
Yeah. Just magnify it. Make it enlarge.
Okay, sir.
No, this
No,
is fine.
just I mean, yeah. Direction is a bit wrong. Yeah.
Okay,
Right.
sir. Then I'll do this way.
Yeah.
Is it fine, sir?
No, now go on
Okay, sir.
higher. It
Good
becomes
morning,
smaller.
everyone.
Yeah.
My name is Dr. Pezangule Chakrunyo. I am from Sasart, Nagaland University. I'm working as a junior scientist in ASRP on soybean. So this morning on poster presentation, I will be speaking on estimation of avoidable yield loss due to diseases in soybean. So this is a part of our trial for this Kharif season. And as you all know, soybean, it is world's most important seed legume. It contributes about 25% of global edible oil, and 2/3 of the protein feed is shared by in the livestock. So it is the feeds are also used for the poultry and fish also, and then mainly in Nagaland, it is used as fermented as akonye. So with this, as you can see in the screen, the world scenario of soybean production, and Nagaland is also producing a large quantity of soybean every year. So this is the data of 2018 to 19. It has been collected from the Department of Agriculture, Nagaland government. So this trial was taken up with an objective to estimate the avoidable yield loss that is caused by the disease in soybean, and then to assess the disease also. So this experiment was conducted in the ASCRP on soybean field at Sasart, Nagaland University, and the variety used was JS9752, which is a 10-year-old variety that was released by the ICR. And then this experiment was laid out in RBD design with 3 replication. So these are the materials and methods that we have used. So for seed treatment and for foliar spray, these are the respective chemicals that we have used. And then we employed 6 treatments. So we have treated with seed treatment and then foliar spray at different days of interval after sowing. So this in this experiment, it was mainly to assess the yield loss and also and side by side to assess the disease incidence and the severity of the disease. So in order to calculate all those, these formulas were used for collecting the data. So in the findings, we in the initial we started with the assessment of diseases. So this Rhizoctonia aerial blight and the next disease, that is rust by Phagospora, and then the pod blight disease. So these 3 are the major diseases that we are facing here in our center. So these diseases mainly, especially the Rhizoctonia aerial blight, it starts from the appearance started from the last week of August, and then the remaining pod blight and the rust disease, they have started from the last week of September. So they're rapid dissemination and the rapid spread of the disease was observed in the month of October 2nd week for rust and pod blight, and then for RAB, for the Rhizoctonia aerial blight, it was observed during the 2nd week, the fortnight of September. So in the assessment of the disease, in the disease incidence, as you can see that the T4, that is the treatment with seed and then seed treatment and then foliar spray, was giving the lowest disease incidence in 3 of the disease, especially in the disease of rust and RAB and pod blight. There was a significant decrease in the incidence due to the application of the foliar spray and the seed treatment. So in the severity also, the T4 performed well, but in pod blight, the T4 treatment was found to, T4 and T3 was found to be statistically at par in case of severity. So with this experiment, with this table, I would like to conclude my presentation with this table. So the avoidable yield loss, that was the data that was collected from the yield, it was directly correlated with the disease incidence and severity. So in this table, we could see that the control plot that yielded only 1,712 kg per hectare, whereas the T4 that was found with the lowest incidence and severity performed well, and the yield was much more higher as compared to the control. And even the average yield was higher. Avoidable yield loss was higher in T4 as compared to control, and as compared to other treatment. Control was 674 as compared to the control. Seed treatment and the use of the foliage. So as field observation, there was a very low density and very low severity of disease found to be in the T4 treatment. So my
hello. So is my network or your network? It's not working. So thank you very much. So hello, it's open to the house for any queries.
Okay, Dr. Chakruno, hello, am I audible to you?
Yes,
okay,
you're audible, sir.
Yeah, you see, we are in a poster session, right?
Yes, sir.
But I don't know why you have not prepared your presentation. That is poster.
Yeah, he has presented in PowerPoint.
PowerPoint. That is my
Yes,
main
sir.
question. Okay, so beside that, what are the main diseases of soybean in the Northeast India?
Sir, it varies from region to region.
Okay.
But our centers are in Johart and then in Mezipema. So in Johart, we have a problem for charcoal rot and YMV.
Okay.
And as far as Nagaland is concerned, rust and pot blight and RAB is more. So in overall, with the assumption of only the two centers, we can say that charcoal rot, YMV, and rust, PB, pot blight, and RAB. So those are the major concern diseases, sir.
So what about the stem rot? Because in long back, it has been reported from Assam that it is a very severe disease, especially in soybean. Around in 1985, it was reported from Assam.
Okay,
Stem
sir.
rot of soybean caused by Rhizoctonia solani.
Yes, sir. Yes, sir. There has been report in this recent years also. There was a report from Johart also, but it was categorized as minor disease from Johart.
Okay. But
during our course of studies, sir.
Yeah, but what about the report I have gone through? They have mentioned it is a major disease, around 30 to 38% loss is there. That is in the early stage of the crop. Okay. Thank you.
Okay,
Thank
sir.
you.
Okay, sir. Yes, sir. Okay, thank you, sir.
Yes, madam.
Yes. I actually wanted to know the charcoal disease you are mentioning. It is seen in the stem, root, or leaf of the soybean plant.
Ma'am, actually, I have not mentioned the charcoal rot in my slides, but if you have asked the question, then charcoal rot is usually present in the stem, ma'am. There can be seen very black small spots on the stems, and then if you split open up, then you can see even inside the tissues also there's black dots will be found in the inside the tissue. Yes.
Causal organism for this is?
Pardon, ma'am?
What is the causal organism pathogen for this?
Ma'am, we have
Charcoal.
not found any charcoal road till here in Sasart. We have not got
You
any
have not.
charcoal road here so far.
Okay, another,
Yeah.
okay, fine. I actually wanted to know, you are mentioning that the soybean variety, which was recently released, not even 10 years, no? Within 10 years, it started showing,
Yes, sir.
developing
Yes,
this
ma'am.
susceptible to the disease. So actually, once the
Yes, yes.
plant breeders release the variety, it should not be susceptible within very short period of time, right?
Yes, ma'am. Yes, ma'am.
So I think if you have your native crop varieties of soybean, because Nagaland is famous
Yes.
for its fermented food products, especially of soybean products, no. So if you have your own that
Yes.
native crop variety, that should be, I think, we should preserve, and then, because the whatever the variety we are getting from ICAR or some from plant breeders, they are showing disease or some kind of, they are not that resilient to that particular in our area. So I think your own native variety of soybean crop can be maybe propagated or conserved in future. That would be nice, right?
Yeah, yes, ma'am. Yes, ma'am. Ma'am, actually, this variety was, it is categorized as moderately resistant for our center.
Okay.
So there has been a very good resistance also, but not to all the diseases, ma'am.
Okay, right. But usually, once we release every aspect, that means resilience, whatever resistance, the variety should be resistant. No, then only
Yes, ma'am.
we scientists they release. But this in
Yes,
this
ma'am.
case, it's not happening. No, it's showing all the diseases. Okay, thank you.
Yes, ma'am.
Sakruno, only one question from my side is very simple. Why the
Okay,
charcoal
sir.
road of soybean is being given the name is charcoal road?
Sir, sir, this is the actually the stem, no, sir. That when in the later stage of the crop, when it's in the senescent stage, so this particular crop is completely turns into black, sir, with covered with the black spots, sir.
Yeah, but why the name has given as charcoal road?
Sir, this
How?
is this may,
It
sir, this
forms
may be,
like charcoal, like
Yeah.
charcoal.
Only that much. I want
yes,
to know
ma'am.
is the appearance look like charcoal. Okay, because of
Yes,
that.
yes, that's what, sir.
Yeah,
Yes.
No,
only
no,
that.
no.
Okay, thank you,
Yeah.
thank you.
Okay, thank you, sir. Thank you for your question, sir.
So now I call upon Purva Sandhya. Purva Sandhya. Hello.
Sir,
Oh,
Chairman, sir.
yes.
She opted for MJ Narasimhan Award presentation.
Yes.
So her abstract will go to the next session. So you
Okay.
can continue from the next speaker, next presenter.
Okay. So then Tampak Lema. Tampak Lema. I think she will not be there. Tampak Lema. So I think she is not there. So I'll go to the last contestant, M. Bora. Hello, Mr. M. Bora. Not there. M, what shall we do? M. Bora also not present. Tampak Lema also not present.
I think we missed two participants who could not present because of poor network. We
Okay.
can try again if they are
Yes.
ready.
Yeah, the previous one.
Yeah, I mean serial number 7, Tianla.
Taylor, you are here.
Yes, sir. Yes, sir.
Yeah. Please carry on. So we were very sorry because of the network. So
Sorry, sir.
we detained you for a long time. Okay. So please proceed.
Okay, sir.
Your
Okay, sir.
presentation.
Good afternoon, everyone. So I'll be presenting on the effects of different substrate pre-treatment methods, the growth and yield of Hypsicus ulmaris. Mushrooms are considered as a healthy food, and its cultivation represents one of the most important processes of organic recycling process. However, substrate used for its cultivation are contaminated with various kinds of microorganisms, which can be detrimental for its growth and production. Therefore, for the optimization of its production, substrate must be subjected to the process of sterilization. Hello, am I? Hello.
Yes, yes, yes.
Okay.
Yes, you are audible.
Oh, okay, ma'am. Okay, ma'am. So therefore, considering everything, I have conducted a research using body straw as my substrate and with 7 different treatments and giving a control. I followed the method described by Banoy and Nagarjan 1976 with 7 different treatments, namely hot water treatment at 80 to 90 degrees Celsius for 30 minutes. And then for treatment 2 and 3, I used ginger extract at 4% and 6% respectively. And for treatment number 4 and 5, I used garlic extract at 4 and 6% respectively. And for treatment number 6 and 7, I used neem extract with 4 and 6% respectively. After conducting my research, I came to the conclusion that hot water treatment at 80 to 90 degrees Celsius for 30 minutes gives significantly better result with higher yield and higher biological efficiency. With that, I conclude that we can go for hot water treatment for the cultivation of Hypsicus ulmaris, keeping in mind that we can also, however, the mechanism behind the better performance of the hot water treatment as compared to other studies can also be looked into for further researches in this line. Thank you.
So any queries open to the house?
Yeah, thank you, Tianla. So I have one simple query.
Yeah, thank you, sir.
Though you have used that hot water treatment, right? So during the cultivation
Yes, sir.
of that species, whether you got any type of contamination with other fungus, especially I have seen while doing work on, say, oyster mushroom, used to get the infection of your trichoderma as well as penicillium in the straw. So in your case, whether you got that type of infection?
No, sir. For hot water treatment, I didn't get any kind of infection, but I couldn't grow any mushroom control. Was largely affected with blue mold.
Blue mold, okay, okay. How you control that one? How could able to manage that blue mold?
For sir control, I didn't use any.
Any
Sir,
botanicals?
for this experiment, I didn't use. Pardon, sir?
Any botanicals you used? No.
No, sir.
Okay, okay. Thank you. Thank you
No,
for your
sir. Because
presentation.
some botanicals has been used as a treatment, sir.
Can you cite example? I could not able to get you actually.
Sir, I use a garlic
Garlic
sir, am I
extract,
audible,
right?
sir?
Yes, yes.
So how
Yes, sir.
you
I have used
incorporated? How you incorporated the garlic?
Garlic at what percentage?
Sir,
You have to spray, or how you?
I use 4% and 6%.
And how you that is spray or incorporated,
Sir,
or what? Pardon?
Sir, for this treatment, I soak the substrate for 16 hours as described by Biswas 2015.
Now, what about garlic extract? How you apply it?
So for all the treatment, I soaked the substrate in the extract at recommended concentration.
Was it benefited? That means what is the cost benefit ratio with the control? Whether you have calculated?
Sir,
Because
I think
the
it will be
garlic
100%
is
because in control.
The cost of garlic is too high. That's why I'm asking about the cost benefit ratio. If you have calculated, can you please tell me?
Yes. Sorry, sir, I didn't calculate the cost benefit ratio, so
Tayana, this is
I can't say
from my
exactly.
side. So
Okay,
your
thank you.
garlic use will be very expensive. So whatever normally what we are spawning bag, it costs completely. I'm altogether for one spawning bag costs 55 rupees.
Yes, sir.
Okay, the profit will be I mean double times or 3 times of that. So if you use garlic, so how much amount of garlic you are going to use for one spawning bag?
Sir, it depends upon the fleshiness of the garlic, whether it's fresh or not. It depends upon it largely, because if it's fresh, we can extract lots of juice from the bulb. But if it's a little dry, then we can extract a lot.
Okay, you just proceed for further study for that also. Okay, for particular one bag, how much we need it for I mean
Sure, sir.
soaking for that. So it is very necessary for example carbendazim, we use it for soaking one overnight along with the this formaldehyde. Instead of that, if it is replaceable by this garlic, then it will be better, isn't it?
Yes, sir. Yes, sir. It will be very good. People
Okay,
are being consuming these days.
सर जी सर सुशांत भी थोड़ा राइज करो ना फ्रॉम योर साइड प्लीज हेलो सर सुशांत so from your side also please arise one question if you have little bit doubt or
Okay,
anything else.
Can I ask one question?
Yes, ma'am. Yes, ma'am.
Okay, because most of the substrate preparation, the routine method is hot water treatment, right? For all the substrate, usually for substrate preparation for the mushroom growing. So hot water treatment is common for all. It's a basic procedure, right? Yes or no?
Yes, ma'am.
It's yes.
Yes, ma'am.
Then in addition to the hot water treatment, you treated the substrate with the botanical, some plant extract, right? So,
ma'am,
so
actually,
what is your
I compared.
compare out of the 3 treatments you have done, which was you have found to be giving a better result? Garlic, ginger, and then
Ma'am,
what else? Only hot water. So I think this hot water treatment is not, it should not be the parameter, because it is the main routine method for preparation of the substrate for growing a mushroom. So you should have compared with the extract, botanical extract or some other thing. But this hot water treatment is, I'm just telling you, it's a basic method for all the substrate, and it's a standardized method. So there is no comparison. You have to follow this method. So maybe
madam,
you have gone for the plant extract treatment.
Madam Linji Wangdi, so it's my submission. So as a I mean as a it is practically used in our what to say lab, that hot water treatment.
Yes,
If
sir.
it is used for commercialized, it is very expensive. So normally we use this thing, carbendazim or any systemic fungicide, as well
Yeah,
as in combination with this thing formaldehyde. But we are going to what to say, I mean replaced by any sort of things. So for study point of view, so hot water treatment is giving knowledge to the students. So it will be very expensive, and it takes more time to prepare the substrate. So I'm just at our CAU, I'm holding this post as this thing. So I'm trying to make out it the easiest way. And for example, some insects are there. So if we buy one piece of insect trap, it's very expensive. So I replace it by using the this thing grease. of those automobile grease, the transparent one. So and we cut the pieces of this thing, polyethylene bag, a very lengthy one. Then we just what to say, smear on that both sides, and it is being traveled easily. Practically, it is being done at my this thing mushroom unit. So like that, we are developing. Okay, thank you, Tadela. So it's my submission. Okay.
Yes, sir.
So,
Thank
Madam,
you, sir.
any question? So, Sir Sushant is not asking any questions. So, I want to hear from you also, sir.
Sir, may I please carry on because
Okay.
One more.
Okay. So, uh, our colleague, where he has gone, sir, yours is being completed.
One more is there, na? Dr.
Yeah,
Shurmina
please,
Devi.
please.
Shurmina, are you ready?
One more is there.
Shurmina? Yeah, Shurmina, serial number your 10.
Number 10.
10, serial number 10.
So, and Miss Urmina Devi, Miss or Madam, so you are assistant professor at the College of Horticulture and Forestry. So please proceed. Surmina Devi, please kindly proceed. Hello? Surmina?
Your network issue is not been sorted out yet.
It's not there.
Yes.
What shall
So
we
I
do?
think we are running out of time also. I think we can just wind up, no?
So that means out of 14, 14, 12 are present. 12 or 11. 12 is not there. Urba Sandhya, Sanjit, and Tapalame is not there, and M. Bora is not there. So it is my request, Madam. Shall I send it out to you by distinct marks? Will you add it, or shall you want to complete your own one, and we have to make out it as soon as possible. We are going to submit it before the validity function.
Okay, I will send you my score to your
Okay.
ID. Right?
And Sir Pranab
Mail
Dattaji,
Within 10 minutes,
you are going to send to me, or shall I send to you?
No, within 10 minutes, you will get my result.
Okay.
Okay.
Hello.
Yes.
So thank you so much, sir.
Yes.
Thank you. Kindly sparing your time. So thank you, Dr. Nongden Khomba Singh Laikambam, convener, associate professor and head, Department of Plant Pathology, CAU Imphal. Thank you, Dr. Pranab Datta, associate professor, School of Crop Protection, CPGS Parapani, Meghalaya. And also thank you to Dr. Lanje Wangdi, associate professor, Department of Botany, Sikkim Government College, Gangtok. So thank you so much for your patient and sincere conduct of the session. And with this, we come to the end of the session, e-poster session. So thank you very much.
Thank you.
Thank you for the opportunity.
Okay, thank you, sir.
Thank you.
Since we are running late because of many technical hitches, we will start the next award contest session now. So for this session, we have 8 contestants. And this session will be convened by Dr. R. Bhagavati as convener. He is the principal scientist and officer in charge, ICR, NRRI, RRL, RRS, Hajo, Kamrup, Assam. And the members of this committee are Dr. Karuna Srivastava, Professor, Department of Forestry, NERIST, Nirjuli, Arunachal Pradesh, and Dr. Pankaj Baiswar, Senior Scientist, Plant Pathology, and In Charge Head, Division of Crop Protection, ICR Research Complex, Northeast and Hill Region, Umiam, Meghalaya. So, without wasting much time, may I request now Dr. R. Bhagavati to kindly take over and conduct the proceedings.
Hello.
Yes,
Am I
sir.
audible?
Yes, sir. You are
Yes, sir.
audible.
Yes, sir.
I'm Dr. Rupankar Bhagavati. I welcome all other colleagues of our empaneled for the judging this contestant, Professor M.J. Narasimha Academic Award contest. The candidates are, I think it is restricted to 8 only, or more participants may add up here.
No, sir. It is 8 for MJ Narasimhan,
Yeah.
and we have 1 for APS travel grant sponsorship.
Right.
That is all.
And my colleague will be Pankaj Baiswar and Madam Karuna Srivastava.
Right.
Welcome, all of you.
Good afternoon. Good afternoon to everyone.
Good afternoon, everyone.
Good
And there
afternoon,
will be only in 3 aspects we'll evaluate their presentations. One is your relevance of the work, next is content, and third is delivery. These are the 3 aspects where we'll look into their presentations and we'll evaluate, and finally we'll make average of all the 3 persons and we'll declare the results. Okay, madam?
Okay, I have one suggestion. If we can fix some marks on these 3 aspects so that we can evaluate means similar marks.
Yeah, 20 marks for each aspect.
Okay, okay, no problem.
Baisar, Pankaj.
Oh, sir, it's okay, sir.
20 marks
Sir,
for,
I can hear you.
yeah. One is your
Yeah, it will
relevance
do, sir.
of the work. Relevance of the work, 20 marks. Content will
Okay,
carry
okay.
20 marks, and delivery will also, mode of delivery and all other presentations will carry 20 marks. Total 60 marks.
Oh, fine, sir. Fine, sir.
You will also evaluate against 60 marks. We will also, madam and myself also do. Then we'll send the evaluation, and then you add up and make average of 3.
Shall we keep some marks on discussion also?
There will be no discussion.
There will be no
Yes,
discussion.
Dr. Sushanta Banik, please announce the rules of this competition, so that it will be it is important to for the contestants also.
Okay, for the contestants, each presentation should be of 15 minutes duration strictly, and one warning sound can be made at the end of 13th minute, so that presenter will have to finish his or her presentation within next 2 minutes. Any presenter crossing the 15-minute limit will be disqualified. So out of these presentations, only 2 contestants will be nominated for a national level contest for this Professor M.J. Narasimhan Academic Award. So if there is more to add, Secretary Sir, can you please?
No, it's fine.
No, there is no question and answer session.
Now, after a presentation, it is only presentation in both the this contest, Narasimhan Award contest, as well as APS Travel Grant contest. There is no any discussion and questionnaire session.
Okay, sir.
And
Yeah,
also
I hope the presenters have got it.
judges marks are final. So that will be the last means evaluation. So there is no in between. So judges marks are final.
Yeah.
Yes, sir. Thank you. Thank you.
Okay.
I hope the presenters have got the information already.
So who will be the timekeeper from your side, Susan?
Okay, sir. Okay, sir. We'll take it.
You give the buzzer at 13 minutes and 15 minutes.
Sure, sir. Sure. Thank you.
Okay, all the best to all the contestants.
Sir, shall we invite the first contestant?
Yes. First contestant can be invited.
So now I would like to invite the first contestant, Miss Aarti Kumari. Her title of presentation is Synthesis and Characterization of Biogenic Silver Nanoparticles. So Miss Aarti, can you hear us? Aarti Kumari?
Yes, sir.
Start presentation.
Please start.
Okay. Sir, shall I start?
Yeah.
Yeah. Thank you, sir.
Yes.
A very good morning to respected chairman, co-chairman, all the dignitaries and participants present over here. My name is Aarti Kumari and I'm going to present my research paper on the topic Synthesis and Characterization of Biogenic Silver Nanoparticles, its assessment as antipathogenic activity against Rhizoctonia solani and Xanthomonas oryzae pathobar oryzae, and toxicity on beneficial microbes and mammalian cells. So starting with, rice is one of the most important staple crop which is produced and consumed worldwide. Rice crop is known to be affected by a number of biotic and abiotic stresses. So the common and devastating diseases of rice includes bacterial leaf blight caused by Xanthomonas oryzae, pathobar oryzae, and sheath blight of rice caused by Rhizoctonia solani. So it is known to cause yield loss of about 12 to 75% by BLB, and sheath blight, it causes about 50% yield loss. However, indiscriminate use of chemical pesticides have led to environmental degradation and development of resistance in plant pathogens. So it emphasizes the need of a suitable alternative. And here comes the role of nanotechnology. Nanotechnology is one of the most advanced science of 21st century. It deals with particles in the range 1 to 100 nanometer in at least one dimension. Nanoparticles can be produced by a number of approaches, but biogenic approach is the most recommended one because of its assured rewards such as cost effectiveness. Secondly, they are environment friendly and are easily scalable. Among all the nanoparticles synthesized and studied so far, silver nanoparticles are one of the most celebrated ones. They are known to possess antimicrobial, antiviral, anticarcinogenic, and then wound healing properties. So HNP are extensively used in various fields such as biomedical, textile, pharmaceuticals, etc. In agriculture, silver nanoparticles have the potential to be used as potential plant health material. AGNPs have been found effective against various pathogens such as Fusarium obsisporum, Sclerotium roxi, etc. In addition to this, silver nanoparticles are known to increase production of secondary metabolites as well as plant growth parameters. Therefore, silver nanoparticles have gained access into our daily life, and we can say that proper understanding of its interaction with silver nanoparticles is very essential. So with this background, following objectives of the study were formulated. First, synthesis and characterization of biogenic silver nanoparticles. Second, assessment of antipathogenic activity of biogenic silver nanoparticles against Rhizoctonia solani and Xanthomonas oryzae pathober oryzae. And then study on toxicity of silver nanoparticles on beneficial microbes and mammalian cells. Starting with synthesis and characterization, at first silver nanoparticles were synthesized biogenically from Trichoderma asperillum. So we observed change in color from dark green to light brown. Thereafter, synthesized AGNPs were characterized using various equipments as shown here. At first, UV spectroscopic analysis was done and we observed characteristic surface plasmon absorbance band at 410 nanometer, which indicated formation of silver nanoparticle. Thereafter, average size of the silver nanoparticles were obtained as 49.20 nanometer and the polydispersity index value was recorded as 4.535, which means that the particles were polydispersed in nature. Thereafter, zeta potential value was recorded as minus 28.4 millivolt, which means that the particles were pretty much stable in nature, and the concentration was recorded as 2.24 into 10 to the 7 particles per ml. Then, to understand various functional groups associated with AGNPs, Fourier transform infrared spectroscopic analysis was done and two characteristic bands were obtained. So these bands were, it corresponds to the bending vibrations of amide 1 and amide 2 bands of protein and stretching vibrations of primary amines. So thereafter, these synthesized AGNPs were used to determine its antipathogenic activity against Rhizoctonia solani and Xanthomonas oryzae pathober oryzae. So we found that the radial growth of Rhizoctonia solani, it declined with the increase in AGNP concentration, and the growth inhibition percent was found to be highest at 200 ppm concentration, that is about 73.39%, and followed by at 200 ppm. So these are some of the graphic evidences. Then we moved on to determine the mode of action of silver nanoparticles on this fungal cell. So here, by using scanning electron microscopy, so we found that silver nanoparticles, it accumulated within the fungal cell, and then localized swellings were also observed, and then it caused bending and it caused breakage. Thereafter, that of the cell was observed. So we can say that Rhizoctonia in case of in Rhizoctonia solani, AGNP induced lysis and death of the cell. Thereafter, effect of silver nanoparticles against two rice diseases were estimated. So the two rice diseases were caused by Rhizoctonia solani and Xanthomonas oryzae pathober oryzae. These are some of the treatment combinations, and observations were recorded on changes in the production of secondary metabolites as indicated here. This is a general view of the experiment. So we found that there was a very much increase in production of secondary metabolites in the plants that were treated with silver nanoparticles and were challenged with pathogen as shown here in the graph, treatment T6 and T7. So this indicates increase in the production of secondary metabolites which are involved in biochemical defense mechanisms in rice. So now, after assessing its antipathogenic activity, we moved on to determine the environmental risk associated with these biosynthesized AGNPs. So for this, study on toxicity of silver nanoparticles on beneficial microbes and mammalian cells were done. So for this, beneficial microbes used were Trichoderma hertzianum, Beauveria bassiana, and Bacillus thuringiensis, and were tested against three different concentrations, that is 50, 100, and 200 ppm. Effect of silver nanoparticles on biomass production of fungal biological control agents were done by mycelial weight inhibition technique. So we found that percent inhibition, it increased with the increase in AGNP concentration in case of both the fungi, and it was found highest at 200 ppm. That is for 31.54% in case of Trichoderma herzianum and 28.40% for Beauveria bassiana. These are some of the photographic evidences. Thereafter, effect of silver nanoparticles on biochemical parameters of these fungal biological control agents were determined by estimating total soluble sugar and total soluble protein. So here also we observe similar trend in the result wherein declining total soluble sugar and total soluble protein was obtained for both fungi with the increase in concentration. Moving on to the third beneficial microbe, that is on Bacillus thuringiensis, as clearly indicated in this photograph, we observe that no inhibition zone was produced at all the tested concentration, which means that Bacillus thuringiensis was completely resistant to silver nanoparticles at all the tested concentrations. Moving on to the next part of our cytotoxicity assay, that is on mammalian cells. So for this study, mammalian cell line used was Verocell. So, but before starting the cytotoxicity assay, we need to ascertain the uptake of silver nanoparticles by Verocells. And it was done by biochemical assay. And we observed result as shown here. Even after 1 hour of exposure, there was significant uptake of silver nanoparticles within the cell. And then it increased with the increase in AGNP concentration and was found to be highest at 200 ppm, that is about 98.70%. Then again, with the increase in time of exposure, that is after 4 hours and 8 hours, there was tremendous increase in the AGNP content within the cells. Then, to visualize the uptake of silver nanoparticles by Verocells, we did cellular fluorescence microscopic study. And here, as shown in these photographs, we observed that intensity of fluorescence emitted by silver nanoparticles entrapped within the Verocells, it increased with the increase in concentration. Thereafter, we did the cytotoxicity evaluation of AGNPs on Verocells. At first, effect of AGNPs on Verocell morphology was done by big GM sustaining. As shown here in these photographs, there was no alteration in cellular morphology at the tested concentrations when compared against control. However, when the same test was done using scanning electron microscope, we observed that at 50 ppm concentration, no disintegration was evident, but at 100 and 200 ppm, cellular disintegrations were seen. Then moving on to the biochemical assay, here the percent viability of Verocell after AZNP exposure was obtained by MTT test. So MTT test, it measures the amount of formazole crystals produced by Verocells after exposure. So percent viability was found to be highest at 50 ppm concentration, that is about 75.268%. Then with the increase in AGNP concentration, percent viability, it declined gradually. When compared this data against the standard cytotoxicity scale, our data, it falls in the range of 80 to 60% cell viability, which means weak cytotoxicity, which indicates that AGNPs, it induces mild cytotoxic effect on mitochondrial metabolism of Verocells. Thereafter, lactate dehydrogenase test was done to determine the LDH leakage from the cellular aberrations, and then percent cytotoxicity was calculated. So, percent cytotoxicity was found to be lowest at 50 ppm, that is about 12.15%, and then it increased with the increase in concentration. So AGNP at 200 ppm, it caused highest percent cytotoxicity, that is about 28.22%. So from our study of Verocells, we can say that biosynthesized AGNP poses low cytotoxic effect on Verocells. Now to summarize the entire study, silver nanoparticles at 200 ppm was found to be effective against the diseases, and the mode of action was silver nanoparticles accumulate within the cells and cause swelling and breakdown, which ultimately leads to cell death. Thereafter, biosynthesized silver nanoparticles, it caused mild toxicity on fungal biological control agents as well as on Vero cells, but against Bacillus thuringiensis, it was found to be completely resistant. While studying on Vero cells, we observed that internalization of silver nanoparticles by Vero cells, it increased in a concentration as well as time-dependent manner. And the viability, it declined gradually as well as the person's cytotoxicity, it increased. But it posed mild stress on Vero cells. To conclude, silver nanoparticles at 200 ppm was found effective against the diseases. Second, green synthesized AGNPs poses mild toxicity on beneficial fungal microorganisms and mammalian cell. And third, bacterial biological control agent, that is Bacillus thuringiensis, was found to be completely resistant to AGNPs. So further, we need to elucidate suitable mechanisms to develop nanoformulations and also to reduce the toxicity of AGNPs. And also we need to develop, so we need to understand the mechanism or to reduce toxicity of biological control agents. Further. Thank you, sir.
Next.
So with the permission of Chairman,
yeah,
may I now invite Dibyashri Datta.
Yeah.
Dibyashri Datta. Her presentation is on brinjal little leaf disease, an emerging threat to brinjal farming in Assam, India.
Chairman, sir.
Dibyashri.
Yes, sir.
Hello, Chairman, sir.
Yes.
If you once read the names in serial order, I think candidates can be prepared accordingly.
Yeah. The list is with us. First one is over, R.T. Kumari. Second is Dibyashri Datta. Then third one will be Kakumoni Saikia. Third one. Fourth will be Vishal Saikia. Fifth will be Gitashri Das. Sixth, Lanmo Gohain. Seventh, Sangita Saikia, Swagata Saikia. And the last one will be Purva Sandhya R. Purva Sandhya R. This is for M.J. Narasimhan Academic Award Contest. These are the total 8. Already
this is for convenience of the presenters.
Yeah. So
thank you.
First one is over. Now second one is your Dibyashri Datta should come and show their presentations. Number 2.
Sir, am I audible?
Yeah. Time starts now. Dibyashri, you start now.
Yes, sir. A very good afternoon to the jury dignitaries and all the participants of IPS Virtual Symposium. My name is Dibyashri Dutt and I'm from College of Agriculture, Assam Agricultural University. Today, I am here to present for Professor M.J. Narasimhan Award my topic, Brinjal Little Leaf Disease, an emerging threat to brinjal farming in Assam, India. So these are the contents. Starting from the introduction. Brinjal, also called as egg, belongs to the family Solanaceae. It is an important vegetable crop grown throughout the year in all parts of India except in higher altitudes. Coming towards the horticultural statistics, in Assam,
brinjal is grown in an area of 17.76 hectare, a productivity of 16.12 metric tons per hectare. Brinjal is affected by several diseases, among which brinjal little leaf disease caused by brinjal little leaf phytoplasma is one of the most important and limiting factors in brinjal cultivation. This disease was first reported in Farm, Coimbatore, India by Thomas and Krishnaswamy in 1939. So coming to the rationale, brinjal little leaf disease is a very serious disease of brinjal causing considerable economic losses. In severe affected plants, it can cause yield loss up to 100%. An incidence of 12.5 to 14.5 was obtained from Bishwanath district of Assam and 3 to 34% was reported from India. Although brinjal little leaf suspected symptoms have been observed from time to time in Assam, but systematic studies have been initiated on it regarding its incidence, yield loss assessment, identification of insect vectors, and its natural spread in Assam. As such, considering the importance and significance of the disease, this present investigation was undertaken to study the brinjal little leaf disease with the following objectives. So my first objective was to study for symptomatology, incidence, identification of vectors of brinjal little leaf disease in Assam, and my second objective was for detection and characterization of brinjal little leaf phytoplasma in Assam. So coming towards the materials and methods of first objective, the survey was conducted in 7 major brinjal growing districts of Assam, namely Bishwanath, Darang, Golaghat, Jorhat, Nagaon, Sibsagar, and Sonitpur. After that, symptomatology was recorded and percent disease incidence was calculated by using the formula given by Sridhar and his co-workers in 2013. Then collection of leafhoppers were done by using SNP method and identification of leafhoppers was done in Department of Entomology, AAU, Jorhat. And then insect transmission test was carried out by using 4 different species of leafhoppers, namely Amrasca bigutula, Ecdanus indicus, Hishimonas phycetes, and Nymphotetis nigropictus. And then the other transmission test was carried out using the protocol given by Marcon and his co-workers in 1997. The effect of brinjal little leaf disease on plant growth and yield parameters were recorded by using the formula given by Pathak in 2011. So coming towards the results and discussion objective 1, various symptoms of brinjal little leaf disease have been observed in different farmers during survey. Brinjal plants infected at earlier growth stages develop symptoms such as reduction in size of leaves, stunting, yellowing of leaves. Here, the reduction in size of leaves is the most characteristic symptom of the disease. After that, infection at later stages resulted in symptoms such as floral viruses, phyllodes, and witch's broom. Infected plants also produce other types of symptoms such as excessive branching, curling and cupping of leaves, reduction in petiole and internode length, mummification of fruits, and it finally leads to necrosis and death of plants. Variants of brinjal little leaf disease was also observed on alternate hosts such as Datura and Solanum violaceum, and this is the first report of phytoplasma disease in Solanum violaceum plant. So coming towards the discussion, similar to the results of present investigation, variation in symptom development with the age of the plant at the time of infection was reported by workers such as McCoy, Bertamini, Samueller, etc. So this table 1 shows incidence of brinjal little leaf disease in farmers' field of Assam. Out of the 7 districts, highest disease incidence was observed in Sibsagar district with mean disease incidence percentage of 23.63% and the lowest disease incidence was observed around district with the mean disease incidence percentage of 3.62%. Now, during the survey, 4 different leafhoppers were found in the brinjal seed. They were Hishimonas phycetes distant, Exitianus indicus, Amrasca bigutula ishida, and Nymphotetis nigropictus tall. So out of the leafhopper species, Hishimonas phycetes was most abundantly available with the mean population of 4.85 numbers, and Nymphotetis nigropictus tall was least abundantly available with the mean population of 2.85 numbers. So this graph shows that the higher population of Hishimonas phycetes in Golaghat and Sibsagar district were positively correlated with higher disease incidence in those districts. So coming to the discussion, similar to the results of present investigation, Kumar and his co-workers in 2017 has observed that the higher population of Hishimoto's phycetes in brinjal fields at Delhi and Noida was also positively correlated with the increasing disease incidence in those areas. So this table shows that out of the 4 leafhoppers used during the transient studies, only the Hishimoto's phycetes was able to transmit the phytoplasma from an infected to a brinjal plant with disease transmission efficiency of 80%, and the time required for first appearance of symptoms was 30 to 35 days. So this picture shows the brinjal plants after transmission with Hishimoto's phycetes, and out of the 5 inoculated plants, 4 became symptomatic, which was further confirmed by PCR analysis. Here, this is the molecular detection of phytoplasma in leaf-populated brinjal plants. So all the symptomatic brinjal plants yielded an expected amplicon size of 1.5 kb from direct PCR and 1.25 kb from nested PCR. Molecular detection of phytoplasma was also done in leafhopper vectors. So out of the 4 leafhopper species, only Hishimoto's phycetes gave a positive result for phytoplasma and gave a band size of 1.5 kb from direct PCR and 1.25 kb from nested PCR. So coming towards the discussion, the results of the present investigation are in conformity with earlier workers such as Anjana Yulu, Srinivasan and Chelia, Azadwaran, Baranwal, etc. And they said that the positive association of Hishimoto's phycetes was positively correlated with the disease. Then table 4 shows the transmission of brinjal little leaf phytoplasma through dodder. Out of the 5 inoculated brinjal plants, 3 were infected with a disease transmission percentage of 60%, and the time required for first appearance of symptoms was 30 to 35 days. So this picture, plate number 24, shows the dodder inoculated brinjal plants, and out of the 5 inoculated brinjal plants, 3 became symptomatic, and this was confirmed by PCR analysis. So in the PCR analysis, we can clearly see that all the symptomatic brinjal plants yielded an expected amplicon size of 1.5 kb direct PCR and 1.25 kb from nested PCR. So coming towards the discussion, the present investigation was in conformity with Ratnama, who also reported the successful transmission of phytoplasma by dodder with a transmission efficiency of 40%. Now coming to this table 5, this says the effect of brinjal little leaf disease on growth parameters of brinjal plants. So when plant infected early, that is 65 to 70 days after transplanting, highest reduction in plant height, leaf size, petiole length and internode length was observed. And when plants are infected in their later stages of growth, then lowest reduction in parameters took place. This table shows the effect of brinjal little leaf disease on yield parameters of brinjal plant. So highest reduction in average number of fruit per plant, weight of fruit, fruit yield per plant was obtained when plants were infected. When plants were infected early, that is 65 to 70 days after transplanting. So as the age of the plant in development of the symptom increased, the effect on yield parameters also reduced significantly. So coming on to the discussion, the present findings are in conformity with Pathak 2011, who reported that plants infected at early stages of growth were affected more as compared to plants infected at later stages of growth. Now coming on to the second objective of materials method. Now plant DNA isolation was done according to the protocol given by Collar and his co-workers in 1990. Insect DNA isolation was done according to the protocol given by Marjachi and co-workers in 1998. PCR analysis was done by using 2 serimer pairs, P1P6N, R16S2N, and R16R2. And then gel electrophoresis was done according to the standard protocol given by Sambrook and Roussel in 2001. Then purification of those PCR products were done by using gel and PCR cleanup kit, and those purified PCR products sent for sequencing in 2 companies, that is Bioserve Biotechnologies India Private Limited and Europe Genomics India Private Limited, and then the phylogenetic tree was constructed using MegaX software. At last, the effect of tetracycline sprays on remission of brinjal little leaf symptoms were carried out on 3 different concentrations, that is 300, 400, and 500 ppm. So moving on to the discussions and results and discussions of objective 2. Now, molecular detection of phytoplasma in brinjal plants. So these are the representative samples of brinjal plants collected from 7 districts of Assam, and we can see that all the symptomatic brinjal plants yielded expected amplicon size of 1.5 kb from direct PCR and 1.2 kb from nested PCR, confirming the presence of phytoplasma in them. Then molecular detection of phytoplasma in all hosts were done, and those were the Tura and Solanum violaceium, and we can clearly see the band size of 1.5 kb from direct PCR and 1.25 kb from nested PCR from both the plants. So this confirms them potential reservoir host of phytoplasma in Assam. So this is the phylogenetic tree constructed based on 16S rDNA, and this shows that the brinjal little leaf phytoplasma under study was associated Candidatus phytoplasma trifoli 16SR6 clover proliferation group. Coming on towards the discussion part, the findings of the present investigation were in conformity with the work of Kumar and his co-workers in 2017, who reported that 16SR6 leaf phytoplasma group is the most widespread infecting brinjal crop in India. Now, coming on to this table 7, this shows the effect of tetracycline sprays on of brinjal little leaf disease. So this table shows that 300 ppm concentration of antibiotic tetracycline would not suppress the disease in both single and double sprays, but 400 and 500 ppm concentration of antibiotic tetracycline hydrochloride could suppress the disease for some days, but cannot cure the disease. So this picture shows the effect of tetracycline sprays on remission of brinjal little leaf disease. So plate number 27 shows the brinjal little leaf infected plants before spraying of antibiotics, and plate number 28 shows the brinjal little leaf infected plants after spraying of antibiotics. So we can clearly see the difference in these pictures. Now coming on towards the discussion, the findings of our present investigation are in similarity with Ratnama, who reports that when plants were treated with tetracycline at the rate of 500 ppm of 1, 2, and 4 sprays at weekly interval, the reappearance of symptoms was again observed in all the treatments. So, moving on to a summary and conclusion, brinjal little leaf disease was prevalent in all 7 districts of Assam, and the disease incidence ranged from 3.62 to 23.3% in different locations. 4 different species of leaf hoppers, those are Bigutula bigutula, Exitianus indicus, Hishimonas phycetus, and Nepotitis nigropistol, were observed during the survey, and out of all, only Hishimonas phycetus was able to split the phytoplasma from infected to healthy brinjal plants with a transmission efficiency of 80%. Brinjal little leaf phytoplasma was also successfully transmitted by dodder from infected to healthy brinjal plants with transmission efficiency of 60%. The phylogenetic tree construct based on 16S rDNA sequence has revealed that the brinjal little leaf phytoplasma under study was associated with Candidatus phytoplasma trifoli clover proliferation group. So brinjal little leaf disease severely affected the and yield parameters of brinjal plants, which varies according to the age of the plant at infection. And at last, studies on effect of tetracycline hydrochloride antibiotic in remission of brinjal little leaf disease symptoms elucidated that the effect was purely temporary remission of symptoms. So with this, I would like to end my presentation. Thank you so much.
Thank you.
Chairman sir.
Yes.
Is the bell audible properly?
Yeah.
Okay,
Clear.
okay. Just to
No
check
problem.
it.
Yeah.
Thank you. Thank you.
Now third presenter will be Kakumoni Saikia. Is she ready?
Yes, sir. Am I audible, sir?
Yeah, you are very much audible. So you can start.
Good morning, everyone. Myself, Kakumuni Saikya. Today, I'm going to deliver my presentation on the topic antibiotic exuding rhizospheric streptomyces for biological management of Xanthomonas oryzae, Petrobor oryzae in rice. We know that rice is one of the staple food crop feeding about half the humanity, but the crop is highly susceptible to various diseases, among which bacterial blight caused by Xanthomonas oryzae, Petrobor oryzae, is one of the most devastating and oldest disease of rice, causing loss of about 50% during conducive conditions. A variety of disease management strategies have been employed with associated limitations. Use of biocontrol agents and plant growth promoting microbes offer an attractive way to manage diseases as well as improve crop growth and development, thereby supplementing the use of fertilizers and pesticides. Now moving on to the importance of my research program, is why did I select this program? So, the success rate of some biocontrol agents under field conditions is limited. Hence, in this study, a new group of biocontrol agents have been ventured for the effective management of bacterial blight of rice. Moreover, various researchers have reported the use of rhizospheric streptomyces for management of plant diseases. Hence, the concept to use rhizospheric streptomyces for biological management of Xanthomonas oryzae, Petrobor oryzae seem to be very interesting. These were the objectives of my research program. Accordingly, bacterial blight-infected rice plants were collected from Regional Agricultural Research Station, Tittabur, Jorhat, and then the pathogen was isolated following standard protocol. This table shows morphological and cultural characteristics of the pathogen Xanthomonas oryzae, Petrobor oryzae. Morphologically, the bacterium was gram-negative and rod-shaped. Moreover, it showed light yellow convex, smooth, opaque colonies on sucrose, peptone agar, and nutrient agar plates. This table shows the biochemical characteristics of the pathogen Xanthomonas oryzae, Petrobor oryzae. This plate shows the pictorial representation of the various biochemical tests that were conducted to characterize the pathogen Xanthomonas oryzae, Petrobor oryzae. This plate shows that the bacterium was gram-negative as it retained red color when counterstained with safranin. The bacterium was also observed under field emission scanning electron microscopy, which revealed the bacterium to be rod-shaped. Now moving on to objective 1, that is isolation of rhizospheric streptomyces of rice plant ecosystem. The rhizospheric streptomyces were isolated following the protocol given by Maliki and his co-workers in the year 2013. The rhizospheric soil were collected from 10 to 50 centimeter depth. Altogether, 16 rhizospheric streptomyces were isolated from different locations, which were assigned with unique identification codes such as S1, S2 up to S16. So all the 16 rhizospheric streptomyces were tested in vitro against Xanthomonas oryzae, Petrobor oryzae following dual culture assay method. And this plate clearly shows that among 16 streptomyces, 2 isolates, that is S2 and S15, could inhibit the pathogen effectively. Further, compatibility test was done among the 2 streptomyces S2 and S15, and we can clearly observe in this plate that both the isolates were compatible with each other. Then, this plate shows that the S2 plus S15 could inhibit the pathogen effectively. So this table clearly shows that S2 plus S15 could inhibit the pathogen up to an extent of 56.22%, followed by S15 and S2. This table shows the morphological and cultural characteristics of the rhizospheric streptomyces S2 and S15. Morphologically, the bacteria were gram-positive and filamentous in shape, and culturally, the bacteria were the S2 was gray in color. That is, the colony of S2 was gray, whereas S15 was dusty pink. This table shows the biochemical characteristics of rhizospheric streptomyces S2 and S15. Here, this plate clearly shows that the S2 and S15 were gram-positive and filamentous in shape. They were also observed under field emission scanning electron microscopy, which revealed the bacteria to be filamentous in shape. Further, genomic DNA of S2 and S15 were subjected to agarose gel electrophoresis, which showed a clean amplification of about 1400 base pair. This is the consensus sequence of rhizospheric streptomyces S2, which was of about 1317 base pair. And phylogenetic analysis revealed that S2 showed highest homology with that of Streptomyces fumigarius.
This is the consensus sequence of rhizospheric streptomyces S15, which was of about 43 base pair, and it showed highest homology with that of Streptomyces lawrencei. This plate shows a pure culture of the rhizospheric Streptomyces S2 and S15 on Kenlite media. Moving on to objective 2, that is combination of rhizospheric Streptomyces for management of bacterial blight of rice. Accordingly, selected rhizospheric Streptomyces were tested under pot condition to test their efficacy in managing bacterial blight of rice by applying a seed treatment, root treatment, soil treatment, and spray application. There were 5 treatment combinations, including 1 treatment as inoculated control and 1 as chemical check. Plate A shows the seed treatment, B root treatment, C soil treatment, and D spray application. This table clearly shows that rhizospheric Streptomyces S2 and S15 could reduce the disease incidence as well as disease severity. The lowest disease incidence and severity was recorded in chemical check, followed by the treatment S2 plus S15. Moreover, highest yield and yield attributing characters were also observed in plants inoculated with rhizospheric Streptomyces S2 and S15. Highest yield was observed in chemical check, and the highest yield attributing characters were observed in plants inoculated with S2 plus S15. Moreover, total phenol contents in rice plants treated with rhizospheric Streptomyces were also estimated using Follinsky culture reagent. The highest phenol content was observed in plants treated with S2 plus S15, which we can clearly observe in this table. The highest phenol content was about 2.52%, followed by plants treated with chemicals. Moving on to the results and discussion, among 16 Streptomyces, 2 S2 and S15 could effectively inhibit the pathogen up to an extent of 56.22%. 16 sRNA sequencing results of S2 and S15 showed maximum homology with that of Streptomyces fumigarius and Streptomyces lawrencei. Park and his co-workers also reported that Streptomyces botryopensis can effectively inhibit Xanthomonas oryzae, Vetiver oryzae. So we can clearly observe that Streptomyces fumigarius plus Streptomyces lawrencei treated plants recorded a significantly lower disease incidence and severity, as well as increase in attributing characters compared to individual treatments. However, highest disease reduction and highest yield was recorded in chemical check over absolute control. So the disease management efficacy of the treatments comprising Streptomyces fumigarius plus Streptomyces lawrencei is near comparable with the results of chemical check. That is, they were statistically at par. Moreover, highest yield attributing characters were recorded in plants treated with Streptomyces fumigarius plus Streptomyces lawrencei. So besides being able to reduce the intensity of the disease, Streptomyces fumigarius and Streptomyces lawrencei also improve the growth of the plants. Moreover, highest phenol content were recorded in plants treated with Streptomyces fumigarius plus Streptomyces lawrencei. So combination bioagents better than the individual bioagent, which might be due to their multiple modes of action as well as synergistic effect of more number of organisms. Now, moving on to the third objective, that is detection of antibiotics produced by rhizospheric Streptomyces using liquid chromatography mass spectrometry. The presence of various peaks that appeared in the chromatogram showed the presence of various compounds in the methanol crude extract of Streptomyces S2 and Streptomyces S15, which we can clearly observe in this figure. This table shows the detection of compounds produced by S2 and S15 using liquid chromatography mass spectrometry analysis. There were 5 compounds which were produced by Streptomyces S2 and there were 4 compounds which was produced by Streptomyces S15. It is important to note that the compound APHC4 was produced by both rhizospheric Streptomyces S2 and S15. Now moving on to the results and discussion, in this present study, 5 compounds in the Streptomyces fumigarius extract and 4 compounds in Streptomyces lawrencei extract were tentatively identified using liquid chromatography mass spectrometry analysis. Kanamycin is an aminoglycoside antibiotic that binds irreversibly to the bacterial ribosomal subunit, causing misleading of tRNA. Moreover, clavulanic acid is an inhibitor of beta-lactamase enzymes. It is responsible for hydrolysis of beta-lactam antibiotics. So, a number of these compounds produced by both Streptomyces fumigarius and Streptomyces lawrencei can lead to direct inhibition of the pathogen Xanthomonas oryzae, Vetiver oryzae. Further, plant growth promoting activities were also evaluated for both rhizospheric Streptomyces S2 and S15. Here we can clearly observe that S2 and S15 showed positive results for hydrogen cyanide production test as the color of the filter paper turned brown. Moreover, S2 and S15 also showed positive results for indolysinic acid production test as the color turned pink. Moreover, S15 showed positive results for phosphorus solubilization tests as clear zones were formed around their colonies when inoculated in Pickovskaya media. And S2 and S15 also showed positive results for zinc solubilization tests as clear zones were formed around their colonies when inoculated in JSB media. S2 and S15 also showed positive results for siderophore production tests as yellow to orange halo was formed around their colonies when inoculated in CAS media. This table shows the plant growth promoting characteristics produced by the promising isolates S2 and S15. Moving on to the results and discussion, S2 and S15 showed positive results for hydrogen cyanide indole acetic acid siderophore production as well as zinc solubilization. Moreover, S15 showed positive results for phosphorus solubilization. These results were confirmatory with the results showed by Anwar and his co-workers in the year 2016. So these results suggest that increased growth of rice plants due to application of the rhizosporic streptomyces, that is Streptomyces fumigarius and Streptomyces laurenti, might be due to the production of indole acetic acid, hydrogen cyanide, siderophore, as well as zinc and phosphorus solubilization. Moving on to the summary and conclusion, Xanthomonas oryzae was isolated from bacterial blight infected rice plants which was collected from Regional Agricultural Research Station Titabar Jorhat. Altogether, 16 rhizosporic streptomyces were isolated, among which S2 and S15 could inhibit the pathogen effectively up to an extent of 56.22%. Further, morphological, biochemical, molecular characterization and field emission scanning electron microscopy were conducted to characterize the effective rhizosporic streptomyces S2 and S15. So the lowest disease incidence and severity were recorded in chemical check, followed by plants treated with Streptomyces fumigarius plus Streptomyces laurenti. They were statistically at par. Highest yield attributing characters were also recorded in plants treated with Streptomyces fumigarius plus Streptomyces laurenti. Highest phenol content was observed in plants treated with Streptomyces fumigarius and Streptomyces laurenti. The antibiotics which were produced by Streptomyces fumigarius and Streptomyces laurenti were also tentatively identified using liquid chromatography, mass spectrometry, which might be responsible for their profound antibacterial activity. So these results indicated that rhizosporic streptomyces, that is Streptomyces fumigarius and Streptomyces laurenti, have great potential to be developed as biocontrol agents for the effective management of bacterial blight of rice. So the use of antagonistic microorganisms like Streptomyces species is an appropriate approach to effectively manage bacterial blight of rice. Now moving on to the future prospects, the defense-related pathways of the plant induced by plant growth promoting activities and secondary metabolites of rhizosporic microbes at molecular level needs further investigation. Moreover, studies should be carried out to evaluate the effect of agrochemicals on rhizosporic microbes. With this, I conclude my presentation with a quote given by George G. That is, research is to see what everybody else has seen and to think what nobody else has thought. Thank you.
Thank you. Hello, Susan.
Yes, sir.
Should we continue?
Yeah.
Is there any?
Yeah, you can continue, sir.
Okay. Then now it will be fourth presenter, Vishal Soikia. Vishal
Yes,
Soikia
sir.
is present?
Yes, sir. Am I audible?
Okay. Yeah. Start.
A very good afternoon to respected chairman, co-chairman, and other dignitary present here. My name is Vishal Soikia, here to presenting my research work on the topic study on fungal disease of Zarbra in Assam. These are my contents. As
Vishal,
Hello?
Vishal,
Can you hear me?
Please put your presentation in full screen mode.
Okay, sir.
Yeah.
Now it's okay?
Yes.
As you all know, floriculture is the branch of horticulture, which is mainly deal with the cultivation of flower crop. Floriculture is the very fastest going sector in northeastern region, including Assam, because of availability of diversity in topography, altitude, congenial agroclimatic condition, fertile soil, and well-distributed rainfall, especially domestic and national market. Flower under commercial cultivation in Assam are marigold, glorious, tuberose, anthurium, zarbera, etc. According to National Horticulture Board from 1819, in India, flower is cultivated in an area of 2.49 lakh hectare with a production of 16.59 lakh tons of loose flower and 4.84 lakh tons of cut flower. According to National Horticulture Board 2018-19, in Assam, flower is cultivated in an area of 5,199 hectare. Zarbra is also known as transfer daisy or African daisy. Among the cut flowers, Zarbra is then in 5th position. It has a wide applicability in flower industry as a cut flower and potted plant. Opportunity and scope for research. Floriculture and ornamental industry is one of the most main sector that can provide boost to agriculture economy in our country. Fungal disease that affect yield and quality of flower has been reported by several researchers. Production and productivity of Zarbra is declined due to different fungal disease. Incidence of foliar pathogen is severe in Zarbra. Different scientists they reported that Zarbra is affected by many fungal diseases like powdery mildew, botrytis blight, alternaria leaf spot, sclerotium rot, root and crown rot. Out of which alternaria leaf spot and botrytis blight was one of the most destructive disease in Assam. Coming to objective, isolation and characterization of most serious fungal pathogen associated with Zarbra. Efficacy of different botanical, biogen, and chemical against most serious fungal pathogen. For first objective, we have done roving survey for most of the serious fungal pathogen of Zarbra grown in both open and protected condition in Upper Assam, Lower Assam, and Hill Zone of Assam. For each zone, we have selected 1 to 2 district with 2 to 3 site location. Here, this is the graphical representation of the survey location that we are selected for collection of disease sample and for assessment of percent disease incidence and percent disease index. Here, this is the different site location along with their GPS coordinate. We have also calculated during the survey disease incidence and percent disease index of different diseases. Based here, this is the score chart that are selected for collection of percent disease incidence and disease index. Based on the result, we have found that alternaria leaf spot causing highest disease incidence, which was followed by botrytis blight, and we have also found that colotrichum leaf spot and fusarium will cause sporadic disease incidence. Here, this is the photographic representation captured during the survey period. Here, this is the different typical symptom of fungal disease of Zarbra. Collection of infected leaf sample for isolation of pathogen in nutrient media followed by pathogenicity test. We have done isolation method given by Ricker 1936. A pathogenicity test of isolate fungus was done by Prinpick and mycelial bead inoculation method given by Rosa et al. 1998. Here, this is the photographic representation of pathogenicity test of Alternaria alternata and Botrytis cinerea on healthy Zarbra plant. Cultural and morphological characteristic of Alternaria alternata. We have observed different parameters like type of growth, colony color, both top view and reverse view, shape of the conidia, color of the conidia, septation, and size of conidia, both length and width. Here, this is the photographic representation of cultural and morphological characteristic of Alternaria alternata. Cultural and morphological characteristics of Botrytis cinerea. We have also observed different parameters like type of growth, colony color, shape of the conidia, resting structure, conidiophore, color of conidia, size of conidia, and both length and width. Here, this is the photographic representation of cultural and morphological characteristics of Botrytis cinerea. Based on this cultural and morphological characteristic, we have observed that both the pathogens are identified as Alternaria alternata and Botrytis cinerea, and further confirmation was done by NCFT, National Center of Fungal Taxonomy in New Delhi. Molecular characteristics. Molecular characterization was carried out by Biosaf Biotechnology Private Limited. Here, this is the consensus sequence of Alternaria alternata produced 395 base pair. Here, this is the NCBI database that's showing the homologous with other Alternaria alternata that are present in NCBI. Here, this is the phylogenetic tree showing the genetic relationship of the fungal pathogens. Here, this is the consensus sequence of Botrytis cinerea showing 807 base pair. This is the nucleotide blast result produced showing more than 90% similarity with the other isolate that are present in NCBI database. This is the phylogenetic tree showing the genetic relationship of the fungal isolate with other isolate that are present in NCBI database. Coming to objective 2, in vitro efficacy of different botanical, bioagent, chemical against fungal pathogens of Zarbera. We have selected 3 different botanical based on their medicinal property: Cucuma longa, Allium sativum, and Azadirachta indica. And preparation of pen extract with cold water was done by standard protocol given by different researchers. Here, this is the photographic representation of different steps that are followed during the preparation of pen extract. Screening of botanical against alternata, and Botrytis cinerea. The selected botanical were screening against both the pathogen at 10% concentration through poison food technique. Here, this is the treatment combination for botanicals. Recording of data. The diameter of colony was measured when mycelium fully covered the control treatment, and percent inhibition of mycelial growth was calculated by formula given by Vincent 1927. Based on the in vitro efficacy test of different botanicals against mycelial growth of alternata, we have found that treatment TT, Allium sativum, showing significantly highest mycelial growth inhibition, 86.66%, which was followed by treatment 8. Here, this is the graphical representation of previous result table. Here, this is the photographic representation of in vitro efficacy of different botanical against mycelial growth of alternata. Based on in vitro efficacy of different botanicals against mycelial growth of Botrytis cinerea, we have found that Allium sativum 10% concentration showing significantly highest mycelial growth inhibition, which was followed by treatment 5. Here, this is the graphical representation of previous result table. Here, this is the photographic representation of different treatment combination against mycelial growth of Botrytis cinerea. In vitro efficacy of different biocontrol agent against alternata, and Botrytis cinerea. We have selected 4 different bioagents such as Trichoderma hygienum, Trichoderma viridis, Trichoderma pseudomonas, and Pseudomonas fluorescens was tested against both the pathogen through royal culture technique. Bioagent used during the this is the photographic representation of different bioagents that are used during the course of study. Here, this is the treatment combination for biocontrol agents. Based on in vitro efficacy of different botanical, different biocontrol agent against mycelial growth of alternata, we have found that Trichoderma hygienum showing significantly highest mycelial growth inhibition, which was followed by treatment 9. Here, this is the graphical representation of previous table. Here, this is the photographic representation of in vitro efficacy of different biocontrol agent against mycelial growth of alternata. Based on in vitro efficacy of different biocontrol agent against mycelial growth of Botrytis cinerea, we have found that Trichoderma hygienum showing significantly highest mycelial growth inhibition, which was followed by treatment 9. Here, this is the in vitro efficacy of different biocontrol agent against mycelial growth of graphical representation of in vitro efficacy of different biocontrol agent against mycelial growth of Botrytis cinerea. Here, this is the photographic representation of in vitro efficacy of different biocontrol agent against Botrytis cinerea. In vitro efficacy of different chemical against alternata, and Botrytis cinerea. We have selected 3 chemicals such as hexaconazole, mancozeb, and carbendazim were taken and tested their efficacy against the pathogen by poison food technique. Here, this is the chemical treatment combination. Based on the result, we have found that hexaconazole 0.1% showing significantly highest mycelial growth inhibition, which was 100%, followed by treatment 3. Which followed by treatment 3 as compared to control without combination with other fungicide. Here, this is the graphical representation of previous table. Here, this is the photographic representation of in vitro efficacy of different chemical against mycelial growth of alternata. Based on in vitro efficacy of different chemical against mycelial growth of Botrytis cinerea, we have found that carbendazim showing significantly highest mycelial growth inhibition, 88%, which was followed by hexaconazole without combination with other fungicide. Here, this is the graphical representation of in vitro efficacy of different chemicals against mycelial growth of Botrytis cinerea. Coming to the next objective, in vivo efficacy of different botanical, bioagent, and chemical against major fungal pathogen of Zarbera. The best botanical, bioagent, and chemical found in vitro were selected for further efficacy tests against both the pathogen, both open and protected condition. Here, this is the experimental details. Observation to be recorded for both potent field condition are disease incidence, percent disease index, percent disease reduction, and plant growth parameter such as sucker per plant, flower per plant, diameter of flower, length of the flower stalk, shelf life, base life, shoot length, root length, and facial and diameter of both shoot and root were recorded. Based on the in vitro efficacy test, the best treatment were selected for in vivo study. The efficacy of botanical, based on the efficacy of botanical, biocontrol agent, and chemical against on percent disease incidence of Alternaria leaf spot in pot condition. We have found that hexaconazole 0.1% showing significantly highest percent disease reduction 82.01%, which was followed by Trichoderma hygiena based bioformulation of trigozole that was 76.76%. Here this is the graphical representation. Based on the efficacy of botanical, bioagent and chemical against 1% disease index of Alternaria leaf spot in pot condition, we have found that hexaconazole 0.1% showing significantly highest percent disease reduction 84.22%, which was followed by Trichoderma hygiena based bioformulation of trigozole 78.23%. Here this is the graphical representation. Based on the efficacy of botanical, biocontrol agent and chemical against 1% disease incidence of botrytis that in pot condition, we have found that carbendazim 0.1% showing significantly highest percent disease reduction 84.14%, which was followed by Trichoderma hygiena based bioformulation of trigozole 73.67%. Here this is the graphical representation. Based on the efficacy of botanical, biocontrol agent and chemical against 1% disease index of botrytis that in pot condition, we have found that carbendazim 0.1% showing 88.9% disease reduction, which was followed by Trichoderma hygiena based bioformulation of trigozole 82.39%. Here this is the graphical representation. Here this is the general view of the experimental area under pot condition. Here this is the in vivo effect of different treatment combination against Alternaria leaf spot under pot condition. Here this is the treatment combination for botrytis blood under pot condition. Evaluation of biocontrol agent, botanical and chemical on plant growth parameter against Alternaria leaf spot and botrytis blood in pot condition. We have found that Trichoderma hygiena based bioformulation of trigozole showing significantly highest number of plant growth parameter, which was followed by hexaconazole 0.1%. In case of botrytis blood also, Trichoderma hygiena based bioformulation showing significantly highest number of plant growth parameters, which was followed by carbendazim 0.1%. Here this is the graphical representation of previous result. Coming to field experiment, here we have found that hexaconazole 0.1% showing significantly highest percent disease reduction 79.40%, which was followed by Trichoderma hygiena based bioformulation of trigozole in Alternaria leaf spot in field condition. This is the graphical representation. In case of percent disease index Alternaria leaf spot, hexaconazole showing significantly highest percent disease reduction 85.59%. Here this is the graphical representation. In case of botrytis blood also, carbendazim showing significantly highest percent disease reduction 81.41%, which is followed by Trichoderma hygiena based bioformulation. This is the graphical representation. In case of botrytis blood, for percent disease index, carbendazim 0.1% showing significantly highest percent disease reduction 84.01%, which is followed by Trichoderma hygiena based bioformulation. In case of goat, here this is the general view of the experimental area. This is the field experiment for Alternaria leaf spot, and this is the field experiment for botrytis blood. In case of growth parameter for Alternaria leaf spot, we have found that Trichoderma hygiena based bioformulation showing highest number of plant growth parameter, which is followed by hexaconazole. Here this is the plant growth parameter against botrytis blood showing Trichoderma hygiena based bioformulation showing highest plant growth parameter, which is followed by carbendazim. This is the graphical representation. Here we have summarized that a random survey is conducted in 3 districts of Assam such as Jorhat, Kamrup and Karbi Anglong of Upper Assam, Lower Assam, Hill Zone of Assam to record the disease incidence of different Zarbera going area of Assam. 4 diseases of Zarbera such as Alternaria leaf spot, botrytis blood, Prototrachum leaf spot and Fusarium were recorded. Based on percent disease incidence and percent disease index of disease, 2 frequently occurring disease such as Alternaria leaf spot and botrytis blood of Zarbera with highest disease incidence and percent disease index were recorded for further study. Based on symptomatology, cultural, morphological, and molecular characterization, the isolated fungal pathogens were identified as Alternaria, Alternata, and botrytis cinerea, were further confirmed by National Center of Fungal Taxonomy, New Delhi. Pathogenicity test was conducted by print pig method and micellar beat inoculation method under polyhost condition. In vitro efficacy test of different botanical singly and in combination against Alternaria, Alternata, and botrytis cinerea revealed that micellar growth inhibition 86.66% and 74.44% was showing. In case of biocontrol agent, Trichoderma hygiena showing significantly highest micellar growth inhibition 76.41% and 81.38%. In case of chemical, hexaconazole showing significantly highest micellar growth inhibition in Alternaria, Alternata, and carbendazim showing significantly highest micellar growth inhibition for botrytis cinerea. For in vivo experiment, trichoderma chemical hexaconazole showing significantly highest percent reduction of disease alternata and carbonism showing lowest disease incidence in case of botrytis cinerea. For growth parameter, Trichoderma hygiene-based bioformulation showing significantly highest growth parameter. Thank you, sir.
Thank you. Next presenter will be Gitastri Das.
Shall I start, sir?
Yes, go ahead.
Yes, please.
Good afternoon, everyone, respected chairman, co-chairman, dignitaries, and all the participants present here. I am Gitasri Das. Today, I am here to give a presentation on the topic effect of biopriming and nanopriming on seeds. Sorry, sir. On effect of biopriming and nanopriming on seed storage, plant vigor, and wilt suppression of chickpea. We all know that seed is a basic and key input for agriculture. Of all crops, pulses have a specific importance for the vegetarian population of our country, as it acts as a source of protein. Among the pulses, chickpea is an important pulse crop. India produces about 150 million tons of food grain per year. This production has been increasing due to the production technology, but losses has remained static at 10%. So it is important to increase the quality of the seed, more particularly in storage. Out of different method, priming is considered to be an important approaches. Further till date, no standard packages for storage of chickpea seeds available. So it is an urgent need to develop the same. With this background, we have set two objectives which are as follows. The effect of biopriming and nanopriming on chickpea seed on storage condition, and number two, effect of seed invigoration with biopriming and nanopriming agent on growth and development of chickpea and suppression of fusarium wilt disease. Chickpea seeds were collected from RARS Silongani Nagaon. Seeds were used for study on storage life as well as pot experiment. On storage life, seeds were bioprimed with Trichoderma aspergillum, Metarhiza menisopli, and Beauveria bassiana, containing solution alone and in combination with each other for 1 hour, 2 hour, and 6 hours. Treated seeds were set dry till moisture content reaches to 9%. Half of the 6 hours prime seeds were shown to the pot condition. Shade dried prime seeds were packed in HDPE semi-permeable bag and stored at a room temperature for 9 months. The physiological and biochemical parameters were observed at 2 months of interval. Same procedures were followed for nanopriming, which was described in the next slide. After collecting the seed, the germination and infection percentage, color, and moisture content were determined. These are the biocontrol agents that were used for biopriming for storage, and these are the nanoparticles used for nanopriming. These are the treatment combination for biopriming in storage condition. These are the physiological parameters and biochemical parameters that were observed at 2 months of storage interval. Seed germination and speed of germinations were determined by standard ISTA protocol. This table shows that germination percentage decreases with increase of storage period. Germination of 1 hour bioprime seed was highest at all the storage period in T8, that is biopriming with both Trichoderma aspergillum and Beauveria bassiana, which was followed by T3. Similar trend was found in case of 2 hours and 6 hours bioprime, 6-hour biopriming. Among the three priming times, 6 hours was found best for 9 months of study, 9 months of storage. Similar trend was also recorded in case of speed of germination, germination index, and root length, shoot length, seedling, then seedling fresh weight, then seedling dry weight, and seedling vigor index. This is the summary table of 6-hour bioprime seed for 9 months of storage on growth parameters. Then the seeds were subjected for biochemical analysis. For estimation of peroxidase activity, the Putter 1974 protocol was followed. The result shows that the peroxidase activity of storage chickpea seed increases with increase of storage period. The highest concentration of peroxidase activity was found in control, while lowest in seed bioprime with Trichoderma asperillum in all the priming time, that is 1 hour, 2 hour, and 6 hours. In case of 6 hours, dose peroxidase activity increases with increase of storage period, but in 9 months the increasing rate was lower as compared to the other priming time. This indicates that due to the priming, firmness of the seed increases, resulting the more storage life. For estimation of amylase activity, Burnfield 1955 protocol was followed. The result shows that the amylase activity was found decreases with increase of storage period. The highest alpha amylase activity was found in Trichoderma asperillum treated seed in all the period in all priming time. In case of 6 hours priming time, though activity decreases with increase of storage period, but in 9 months the decreasing rate was lower as compared to the other priming time. For estimation of a total soluble sugar, Yams and Wheels protocol was followed. The results showed that total soluble sugar increases with increase of storage period. The highest concentration of TSS was found in control, while lowest in seed bioprime with Trichoderma asperillum. But in case of 6 hours priming seed, though TSS increases with increase of storage period, but in 9 months the increasing rate was lower as compared to the other priming time. For estimation of total protein, the Loris method were followed. For these, the result shows that the total protein was found decreasing with increase of storage period. The highest total protein activity was found in Trichoderma asperillum treated seed in all the storage period in all priming time. In case of 6 hours, though the activity decreases with increase of storage period, but in 9 months the decreasing rate was lower as compared to the other priming due to the reduction of concentration of the protein that leads to the increase in TSS. This table shows the effect of bioprime seed in storage condition on plant growth parameters and biochemical activity. Biopriming with Trichoderma asperillum from 6 hours has highest growth parameters and biochemical activity, which increases the storage life of a chickpea seed up to 4 months. Then these are the treatment combination for nanopriming in storage condition. The physiological parameters and these are the physiological and biochemical parameters that were recorded. This table shows that the germination percentage decreases with increase of storage period. Germination of 1 hour nanoprime seeds was highest at all the storage in T3, that is nanopriming seeds, nanopriming with zinc oxide nanoparticle, and which was followed by T5, that is the nanopriming with both zinc oxide nanoparticle and silver nanoparticle. Similar trend was found in 2 hours and 6 hours of nanopriming. Among the three priming times, 6 hours was found best for 9 months of storage. Similar trend was recorded in case of speed of germination then germination index, root length, shoot length, seedling fresh weight, seedling dry weight, then seedling vigor index. This is the summary table of 6 hours nanoprime seed for 9 months of storage on growth parameters. Then the seeds were subjected for biochemical analysis. This table shows the peroxidase activity increases with increase of storage period. Peroxidase activity was recorded highest in case of control, but rate of increase was found comparatively lower when seeds were primed with zinc oxide nanoparticle in all the seeds of storage for 1 hour, 2 hour, and 6 hours. This table shows that alpha amylase activity decreases with increase of storage period. Highest concentration was recorded when seeds were treated with zinc oxide nanoparticle in all the periods of storage for 1 hour, 2 hour, and 6 hours. This table shows that the TSS increases with increase of storage period. Highest TSS was recorded in control, while lowest in nanopriming with zinc oxide in all the storage period, that is 1 hour, 2 hour, and 6 hour. This table shows that total protein decreases with increase of decreases with increase of storage period. The highest concentration of total protein was recorded where zinc oxide nanoparticle was used as nanopriming agent in all the priming time. That is 1 hour, 2 hours, and 6 hours. This table shows the effect of nanoprime seed in storage condition on growth parameters and biochemical activity. Nanopriming with zinc oxide has highest growth parameters and biochemical activity, which increases the storage life of a chickpea seed up to 9 months. Now coming to the objective 2, that is the effect of seed invigoration with biopriming and nanopriming agent on growth and development of chickpea and suppression of fusarium wilt disease. These are the biocontrol agents and biocontrol agents used for biopriming against Fusarium oxysporum, Forma species caesarea, and this is the nanoparticles that are used for nanopriming. These are the treatment combination for biopriming, and these are the treatment combination for nanopriming in pot condition. These are the observation recorded, that is the physiological parameters: seed germination percentage, shoot length, root length, number of branches, fresh and dry weight, and seedling vigor index was recorded, and the seed production, number of pods per plant, seed per pod, seed yield per plant, and 1,000 seed weight were recorded. This table shows the effect of biopriming on physiological parameters of chickpea in suppression of wilt disease. Here we found that the seeds which were treated with Trichoderma aspergillum were found highest in physiological parameter, that was followed by the T9. This is a general view of the experiment. This photograph shows the effect of biopriming on plant growth against Fusarium wilt of chickpea. And this shows the effect of biopriming on root growth against Fusarium wilt. This table shows the seed production of chickpea seed. Here we found that the seed that were bioprimed with both Beauveria bassiana and Trichoderma aspergillum shows the highest seed production, which was followed by T4. And this table shows the effect of nanopriming on physiological parameters of chickpea in suppression of wilt disease. Here we found that the seeds that were treated with zinc oxide nanoparticle has the highest physiological parameters, which was followed by T6 that were treated with both zinc oxide and silver nanoparticle. This is a general view of the experiment, and this shows the effect of nanopriming on plant growth against Fusarium. And this is the effect of nanopriming on root growth against Fusarium wilt of chickpea. And this table shows the effect. This table shows the seed production, where T4, that is nanopriming with zinc oxide nanoparticle, has the highest seed production. Moving to the summary, seed germination percentage, germination index, seed speed of germination, seedling vigor index, and fresh and dry weight was observed decreasing with increasing storage time in all the treatment. Biopriming with Trichoderma aspergillum for 6 hours was found in increasing storage life of chickpea seed with germination percentage, germination index, speed of germination, and plant growth parameters. Zinc oxide nanoparticle at the rate of 100 ppm shows the highest seedling fresh weight and dry weight and root and shoot length. Result on POX activity so that increase of storage period POX concentration was found increases, but the rate of concentration is comparatively low. In case of alpha amylase and total protein, there was total protein the lowest concentration was recorded in control but seed bioprimed with Trichoderma aspergillum and nanoprimed with zinc oxide nanoparticles showed the highest concentration of alpha amylase activity. In pot condition. In pot condition, highest shoot length, root length branches were found in T9 followed by T4. In case of nanopriming, the highest physiological parameters were recorded in zinc oxide nanoparticle treated seed. Coming to the conclusion, the present study showed that biopriming with Trichoderma aspergillum and Beauveria bassiana for 6 hours can enhance the life of a chickpea seed up to 9 months with enhanced plant stand and growth parameters with positive effect of biochemical activity. Further trial with the present result may pave the way of storage of chickpea seed by the farming community. Moving to the future trust, more researches should be done on further increase of storage life of chickpea seed. Thank you, sir.
Am I audible, sir?
Yeah, you are audible. Pankaj?
Sir, sir.
So we will continue, na?
Yes, sir.
Sushant will keep the time. Sushant is here or not?
Yes, sir. I will keep the time.
Okay. Lanmo, you start.
Yes, sir. Good afternoon to the respected chairman, jury members, and everyone present here. Today, I, Lonmo Guhai, is here to present on my research title, Detection of papaya ring spot virus in some districts of Assam. So these are the contents for my presentation. Papaya. So papaya is additionally also known as pawpaw or papao, and it is mostly cultivated in all the tropic and subtropic regions of the world. And it is one of the foremost economical fruit crop cultivated throughout the world. Geographically, papaya originated from southern Mexico and Costa Rica. So, and it is a great source of papain, which is also used as a meat tenderizing agent for beauty products, cosmetic products, and etc. So coming to the horticulture statistics, total papaya annual world production is estimated to be 13 million metric tons, where India leads with an annual output of 6 million metric tons and with an area of 97.7 thousand hectares. And in Assam, the area of production is 9.45 thousand hectares. Production is 190 metric tons. So the virus papaya ring spot is first coined by Jensen in 1947 and to describe the papaya disease in Hawaii. It is considered as is one of the most devastating and serious disease of papaya, leading loss up to 100%. The virus falls under the group 4 single-stranded positive sense RNA, family Pochiviridae, genus Potivirus, and it is mostly transmitted by aphid species. This is Major persici, aphid gossypi, and aphid cursivora. The virus is a non-enveloped fleshless rod particle that is between 760 to 800 nanometer long and is 12 nanometer in diameter. So rationally, as there has been consistent report for the papaya ring spot virus from the farmer's field causing yield loss up to 100%, reported by Singh and his co-workers in 2013. However, incidence of papaya ring spot virus was recorded by Chalukdar in the year 2011 in Assam. So the incidence of papaya ring spot virus is much needed to study. The incidence in the following years. Therefore, to fulfill the following research gap, the following objectives are taken. Objective 1: detection and incidence of the papaya ring spot virus in some districts of Assam. Molecular characterization of papaya ring spot virus. Materials and methods. A survey was conducted in the Jorhat, Tinsukia, and Dibrugarh, Majuli districts of Assam during 2018 to 2020. Data was recorded on the symptoms associated with papaya ring spot virus, variety of the crop, rate of severely infected plant. Plants showing characteristic symptoms were collected from the field for further studies. For assessment of papaya ring spot virus disease incidence, total number of papaya plants were counted from the field, and the number of plants showing the characteristic symptoms were also counted and recorded. The percent disease incidence was calculated by using the formula given by Kestrel in 2015. The percent disease incidence was calculated by number of infected plants by total number of plants observed by 100. Molecular analysis of papaya ring spot virus. The samples collected from the field were then tested in the laboratory following the standard protocol of triazole method given by Akkad and Kosnek in 2002. cDNA analysis. After the total RNA isolation, cDNA was prepared using the cDNA kit by using the following ingredients. Then after cDNA, a mixture of 12 microliter was prepared using the following ingredients with a specific primer, and these are the primer details which were used for PCR. This is the PCR cycle with a temperature of initial denaturation temperature, annealing temperature at 50 degrees centigrade at 1 minute. Then after PCR application, PCR cycle, further gel electrophoresis was carried out in the laboratory. Then coming to the results and discussion, symptoms were observed in the papaya field where chlorosis, mosaic, shoestring, distortion of fruits, filiformy, oily streaks on petiole, stunted growth, puckering, ring spots on fruits were also observed. These are the symptoms that were observed during the survey: puckering, mosaic, then filiformy, chlorosis. This is the comparison between healthy leaf and infected leaf, shoestring and blistering, and oily streaks on petiole, malformation of fruit, ring spot fruits. This is a general view of the papaya orchard when clearing. Incidence of severely infected plant. Then here in this table, the severely infected plant were calculated, and it was found that under Jorhat district, Sekoria region had the highest severely infected plant, showing up to 82% disease incidence. Then coming to the disease incidence of the papaya ring spot virus in surveyed field, here Jorhat district showed highest mean incidence, that is 81.7%, followed by Majuli, then Dibrugarh, and then Tinsukia district. Results. During the survey, most of the papaya plants showed mosaic symptoms, chlorosis and mottling. Early symptoms appears as yellowing, vein clearing of young leaves, followed by top leaves of the diseased plant as are much reduced in size. And in the initial fruit symptoms include lightly raised circular spots on the surface, and as the fruit matures, these rings become orange to brown. Other symptoms include puckering, leaf distortion, stunted growth, and infected fruit generally remains smaller, deformed, and the quality of the fruit is also affected. Then highest incidence was observed in Jorhat district, followed by Majuli, Dibrugarh, and Tinsukia. Molecular detection of the papaya ring spot virus through RT-PCR. And here it was seen that the Sequoia gauss showed 100% PRSV infection, where all the samples tested positive for the virus. This is the agarose gel documentation showing 300 base pair product size. Results. Molecular detection of all the surveyed location was done. The samples subjected to RT-PCR showed band up to 300 base pair on gel. The infection percentage ranged from 100% to 57%, and out of 69 samples tested, 50 samples were tested positive for papaya ring spot virus. Objective 2, molecular characterization of papaya ring spot virus. The PCR product that was obtained was purified and was sent to Eurofins Genomics India Private Limited along with the primer pair in a volume of 3 microliter for sequencing. Sequences assembling and context was created using bioedit software. Then similarity of the sequences were checked with other isolate using NCBI BLAST, and phylogenetic tree was created using MegaX software. Results. This table shows the comparison of the papaya ring spot virus with the other viruses found in the other parts of the world. And here Assam isolate shows much similarity with the Jorhat isolate, that is 96 to 80.87%. This is the phylogenetic tree that was created using MegaX software. Summary and conclusion. PRSB was prevalent in all the surveyed districts, that is Jorhat, Dibrugarh, Tinsukia, and Majuli, and the incidence range from 48.75 to 81.7%. The survey results indicated that the symptoms were moderate to severe intensity in older plants. Older plants were as mild in some orchards. Moderate to severe form was observed both in medium to higher age group of plants. Symptoms observed were chlorosis, mosaic, mottling, leaf distortion, stunted growth, filiform shoestring leaves, malformation of fruit, oily streaks on petioles and fruits. All the symptomatic plants that were collected from the districts of Assam yielded the expected amplification. 10 samples tested, 50 samples were tested positive. Blast analysis of code protein genome sequence of the papaya ring spot virus identified in the papaya plants showed similarity up to 89 to 97% identities with other PRSB isolates found in the other parts of the world. The phylogenetic tree constructed based on the code protein sequence has revealed that PRSB under the study is closely associated with the Assam isolate. So papaya ring spot virus severely affects the growth and development of the plants as it reduces the quality and quantity of the fruit in severe cases. It can cause loss up to 800% to the growers under severe conditions. With this, I conclude my presentation. Thank you.
Or you can tell them there also. Sorry, sir.
Hello. Dr. Pankaj.
Yes, yes.
Our chairman, sir, has been disconnected because his computer laptop is giving some problem. He is connecting soon. So we can continue with the next speaker.
Oh, sure, sure. So may I now invite Mr. Swagat Saikia? Swagat Saikia, are you
Yes,
here?
sir. Yes, sir.
Swagat, you can start your presentation, please.
Okay, thank you, sir. So, a very good afternoon to one and all present here, respected dignitaries and jury members. I, Swagata Saikia, a PhD scholar of Assam Agriculture University, Jorhat, is here with my presentation entitled Induction of Defense-Related Biochemicals in Citrus Plant by Microbial Bioformulations and Management of Citrus Canker in Assam. So as we all know that citrus is a genus of the flowering tree and shrub under the family Rutaceae and subfamily Orientidae. The citrus has been affected with a large amount of fungal, viral, and bacterial diseases. Out of it, citrus canker caused by Gentleman's citri, Petavarro's citri, is a most prominent hindrance that causes the citrus canker. A lot of chemical pesticides and antibiotics has been advocated for control of citrus canker, but since it is hazardous to the environment, so agriculture scientists are trying to shift their research and control the disease in an eco-friendly manner. Microbial bioformulations have proved to be a good option in this regard. Keeping this in view, the present study was formulated with two objectives. Objective number 1, efficacy of microbial bioformulation in suppression of citrus canker disease. Coming to objective number 2, assessment of defense-related biochemical changes in citrus plant attributed to application of microbial bioformulations. Objective number 1, that is efficacy of microbial bioformulation in suppression of citrus canker disease. For this, the isolation of the pathogen was done following standard protocol. Identification and characterization of the pathogen was done following standard procedures. This were the biochemical test performed. It was followed by molecular sequencing of the pathogen. Electron microscopy of the bacterial isolate was also done. Immediately, it was followed by checking the in vitro efficacy test of the microbial bioformulations on suppression of XCC. For this, already prepared microbial bioformulations, that is Biosona containing Beauveria bassiana, Biovir containing Trichoderma viride, and Biogreen combination of Trichoderma viride, Pseudomonas fluorescens, Bacillus thuringiensis, Beauveria bassiana, and Metahirsum inosopli was collected from the Biocontrol Laboratory, Department of Plant Pathology, AU Jorhat. Inhibitory effect of 5 bioagent present on the microbial bioformulation was checked on XCC by following the dual culture method. Results revealed that Trichoderma viride followed by Pseudomonas fluorescens and Metahirsum inosopli could inhibit the pathogen in vitro. This were the pictorial representation. It was then followed by checking the field efficacy of these microbial bioformulations on suppression of citrus canker caused by XCC. For this, soil application and foliar spray of the microbial bioformulation was done twice at a month at an interval of 15 days. This was the layout of the plot. This table revealed the incidence and reduction of citrus canker, leaf miner incidence, and yield enhancement after 12 spray of bioformulations at 15 days interval. Interestingly, it was shown that Biogreen, when applied both as soil and foliar application, has seemed to have reduced the disease from 72.99% to 20.96%, with a reduction of the leaf miner from 70.35% to 25.08%, and the yield seemed to have increased from 17.43% to 41.93%. This is the graphical representation of the efficacy of microbial bioformulations on incidence and reduction of canker, leaf miner incidence and suppression, and yield enhancement. Then phyloplane and rhizosporic microbes were isolated following some standard procedures. A total of 5 bacterial and several fungal microbes were isolated, out of which only 4 microbes, that is 2 from phyloplane and 2 from rhizosphere, seem to have proved its efficacy against XCC in vitro conditions. So these were further taken for identification and characterization. This is the pure culture of the phyloplane microbes, and this were the pure culture of the rhizosporic microbes. These were the microscopic observation of the phyloplane microbes, and similarly, these were the ones for rhizosporic microbes. When molecular sequencing was performed, it has shown that the phyloplane microbe P1 to be Geotrichum candidum and P2 to be Cladosporium cladosporoides, R1 to be Trichoderma hergianum, and R2 to be Trichoderma viride. Population dynamics of this phyloplane and rhizosporic microbes were checked before and after application of the bioformulation. It was seen that the microbial dynamics of these biogens have seemed to have increased in those plants where biogreen was applied both as soil and foliar application, followed by biogreen, which was applied only as foliar spray. Coming to objective number 2, assessment of defense-related biochemical changes in citrus plant attributed to application of micro-based bioformulation. For this, secondary metabolites, that is phenolics and defense enzyme, were assayed. Starting with determination of total phenol content of citrus leaf, it was assayed following a standard protocol. It was seen that plants where biogreen was applied both as soil and foliar applications seemed to have the highest phenol content of 7.84%, followed by biogreen, which was applied only as foliar spray. Similarly, assessment of defense enzyme, that is peroxidase, polyphenol oxidase, phenylalanine ammonia lyase, and beta-1,3-glucanase enzyme have shown that in plants where biogreen was applied both as soil and foliar applications seem to have increased gradually from 0 to 24-48 hours with a peak at 72 hours, which then later on declined in the 96 hours. This is the graphical representation of the peroxidase activity in citrus leaf at different intervals after application of the microbial bioformulation. Similarly, similar trend was also followed by polyphenol oxidase enzyme, where it also seemed to have increased gradually from 0 to 24 to 48 hours with its peak at 72 hours, which then declined at 96 hours in those plants where biogreen was applied both as soil and foliar application. This is the graphical representation of the enzyme activity. Phenylalanine ammonia lyase, when was assayed, also have seemed to show the similar results in those plants where biogreen was applied both as soil and foliar application with its peak at 72 hours. At last, beta-1,3-glucanase enzyme, when was assayed colorimetrically, have shown that the biogreen, which was applied both as soil and foliar application, seemed to have gradually increased the enzyme activity from 0 to 24 to 48 hours with its peak at 72 hours, which then declined in 96 hours. This is the enzyme activity. Coming to results and discussion, commercial use of micro-based bioformulation for control of various fungal and bacterial pathogen has already been exploited by Nan Kumar and his co-workers in 2001. Microbial bioagents, Trichoderma viride, Pseudomonas fluorescens, and Metarhizium inisopli present in the microbial bioformulations could highly suppress the pathogen ex situ in vitro. Dual culture assays showed that highest suppression was exhibited by Trichoderma viride, followed by Pseudomonas fluorescens and Metarhizium inisopli. Bazikar and his co-workers in 2016 reported that Bacillus subtilis was effective with inhibition zone of 16.16 mm, followed by Pseudomonas fluorescens 14.63 mm against citrus canker. Biogreen 5 applied both as soil treatment and foliar spray could show highest suppression of canker incidence from 71.28% to 21.05%, leaf minor from 62.02% to 21.49%, and fruit yield enhanced from 17.43% to 41.93%. Das and his co-workers in 2014 used Bacillus subtilis spray suspension in field for significant suppression of citrus canker in Itzid lime. Sarif and his co-workers in 2010 reported that the destruction isolated from 5 isolates of Metarhizium inisopli caused mortality of citrus leaf minor between 30 to 65%. Enhanced dominance of the population count of characteristic and cosmopolitan flora of microorganisms was recorded both in phylloplane as well as in rhizosphere of citrus with application of microbial bioformulations. Efficacy of isolated phylloplane rhizospheric microbes were also tested against ex situ in vitro. Effective ones were identified as Geotrichum candidum and Cladosporium cladosporoides from phylloplane and Trichoderma viride and Trichoderma harzianum from rhizosphere. Similar results were also shown by Andrews in 1992, where he has seen that population dynamics of both phylloplane and rhizosphere seem to have increased after application of PGPR. Abdul Fattah and his co-workers in 2016 was of the view that Cladosporium cladosporoides is one of the most exclusive microbes with antagonistic ability found in the citrus leaf. Mara and his co-workers in 2006 reported that Trichoderma harzianum present in the citrus rhizosphere helps in defense-related gene expression in transgenic lemon plants. The total phenol content was recorded highest in plants treated with biogreen 5 at the rate of 0.2%, followed by BioVir and Biosona. Enzyme concentration was found highest at 72 hours for peroxidase, followed by phenylalanine ammonia lyase, polyphenol oxidase, and beta-1,3-glucanase in plants treated with biogreen under field condition. Phytohormone-treated citrus leaves showed highest level of phenylalanine ammonialized activity and total phenolic compound as compared to control in citrus plant, as shown by Nayam and his co-workers in 2017. Wang and his co-workers in 2008 reported that pseudomonas fluorescens resulted in activity peak of beta-1,3-glucanase and peroxidase and phenylalanine ammonialized against green mold on citrus, which reduced the disease incidence to 21.2% than the control. Coming to summary and conclusion, cultural, morphological, biochemical, molecular, electron microscopy, and pathogenicity studies were conducted to characterize the pathogen. Efficacy of 3 microbe-based bioformulations, that is BioGreen, BioSona, and BioVir, were checked both in vitro and in vivo conditions. In vitro studies revealed that Trichoderma viride was the most effective, followed by Pseudomonas fluorescens and Metrorrhizum inisopli. BioGreen, a combination of Trichoderma viride, Pseudomonas fluorescens, Metrorrhizum inisopli, Bacillus thuringiensis, and Beauveria bassiana, when used both as foliar spray and soil application, showed significant reduction of canker incidence, leaf miner infestation, and enhanced biochemical constituents in citrus leaf, and also increased yield of the fruits. In vitro efficacy of microbes isolated from phylloplane and rhizosphere were checked and molecular characterization was done. Phylloplane microbes was characterized as Geotrichum candidum, Cladosporium cladosporidis, and rhizospheric ones as Trichoderma hargianum and Trichoderma viride. Activity peak of antioxidant enzyme, this phenylalanine ammonialized, peroxidase, polyphenol oxidase, and beta-1,3-glucanase, and total phenol content were seen to be higher in plants treated with BioGreen, followed by BioVir and BioSona as compared to control. Coming to future prospect, research needs to be done with sophisticated technologies to study the changes in defense enzyme activity, which is induced by application of microbial bioformulations, resulting in its regulation. Effectiveness of the consortia used in this study and role of defense-related plant enzyme needs to be checked against all strains of XCC. More FLDs and OFTs needs to be conducted for studying the efficacy of microbial bioformulations against various disease and insect pests. Frequent application of these bioformulations are highly appreciated. With this, I would like to end up my today's presentation with a quote by Albert Einstein, that is, if we knew what it was we were doing, it would not be called research, would it? Thank you all for your patience hearing.
So thank you, Swagata. So may I now invite Purva Sandhya? Purva Sandhya, are you here?
Yes, sir.
So please start your presentation.
Is this screen visible, sir? Hello?
It's visible. It's visible.
Yes, sir. Shall I start now?
Yeah, you can start.
Good afternoon, everyone listening here. I'm Purva Sandhya, MSc Agriculture in Department of Plant Pathology at College of Agriculture, Central Agriculture University, Imphal. Here I'm here to present my research paper entitled Morphochemical Molecular Characterization and In Vitro Management of Pathogen associated with pigeon pea wilt in Manipur. Pigeon pea is a major pulse crop grown in tropical and subtropical countries. And of course, India is the largest producer of it. In Northeast India, it is often cultivated in the perennial forms. In India, the growth rate of pigeon pea is low, and it is influenced by various biotic and abiotic stresses. A large number of pathogens infect this crop, but some are economically important as they cause heavy losses. Fusarium wilt comes under this category, and it is the most economically important and widely distributed. In Manipur, very less or no research has been done on Fusarium. The present work has been designed with the following objectives. First, to study the morphological, chemical, and molecular characteristics of the pathogen. Second, to find out the in vitro efficacy of some agrochemicals, plant extracts, and biocontrol agents on the Fusarium, and also to find out the in vivo efficacy of biocontrol agents on Fusarium. This is my technical plan of work. First, I have conducted the survey and collected the pigeon pea wilt infected samples. Then I have gone for the isolation of the fungus, and then identification of the pathogen was done through morphological, chemical, and molecular characterization. And finally, I have studied the in vitro and in vivo management of the pathogen.
First, I have collected the pigeon pea wilt infected samples. The pigeon pea wilt infected samples show symptoms like gradual withering and wilting of the plants, which results in the dropping and premature drying of the leaves, stem, branches, and finally death of the plant occurs. The main characteristic symptom of this disease is that the yellowing of leaves is seen, and browning or blackening of xylem vessels or vascular bundles is also seen. And also you can see the purple band that extend upwards from the base of the stem. That may be absent in the end plants, but the blackening of the stem is obvious there. After collecting the samples, I have gone for the isolation of the pathogen, and I have obtained the pure culture by Haifel tip culture method and maintained as slants at 4 degrees Celsius. After the isolation, for confirmation of the pathogen, I have conducted the pathogenity test by using soil inoculation method, and while re-isolating the pathogen, the same fungus was found, and hence it proved the Coates phosphate. After confirmation, the morphological characteristics of the pathogen was studied at IARI New Delhi. The pathogen was observed at 10x and 40x magnification, and this image, the microscopic observations revealed that the pathogen shows the produces macroconidia, microconidia, and chlamydospores. In this image, you can see the microconidia and macroconidia both are hyaline, and microconidia is small, elliptical, with singular two cell, and macroconidia are linear, curved, and with 3 to 4 septa, and maybe pointed at both ends. And I also measured the size of conidia using MICAP software at IIA New Delhi, in which microconidia measured 2.1 to 5.1 by 1.1 to 2.2 micrometer, and macroconidia measured 17.1 to 20.2 by 3.0 to 4.1 micrometer. Based on the principles given in the book of Quebec and Harman, the fungus was morphologically identified as Fusarium oxysporum, former species Udam. And after morphological characterization, I have gone through the molecular characterization of my pathogen. First, I have isolated the DNA, and then the isolated DNA was amplified by using PCR, using IPS1 and IPS4 primers. Then the gel electrophoresis was carried out for the amplified DNA with using ethidium bromide, and the final PCR product was sent to Bangalore for sequencing, and the obtained sequence was analyzed by NCBI NBLAST program, and finally the gene sequence was submitted to the gene bank and obtained accession number as MN503267. And then this is the gel doc image of my pathogen's DNA, and the phylogenetic relationship is given here. The phylogenetic neighbors were determined from the nucleotide database of NCBI using NBLAST. The phylogenetic sequence was also analyzed using MEGA7 software, and the phylogenetic tree was constructed. And after the morphological and molecular characterization, I have carried out my chemical characterization of my pathogen at the Tamil Nadu Agriculture University, Coimbatore. Here, the GCMS analysis was carried out in methanolic extract of Fusarium udum. This figure, the figure shown in the slide, that is GCMS chromatogram of my pathogen, shows the presence of bioactive compound. Among these compounds detected here, I have selected 10 compounds based on the relative abundance of the peaks, with the range from 18.55 to 29.14. The compounds identified here was confirmed with the National Institute of Standards and Technology. After this, to pursue the suitable culture media for the mycelial growth and sporulation of Fusarium udum, I have taken used 10 different media, in which potato dextrozagar media shows highest maximum mycelial growth. And then while studying the mycelial characteristics, in this 10 different media, the mycelial color varies from white to light brown, and the pigmentation gets darkens with the age, and the texture is fluffy and regular, and excellent sporulation was observed in potato dextrose agar media, followed by Zapac stalks and sabots. And later, the growth and sporulation of Fusarium udum was analyzed at different pH levels, and significant variation is seen here. The growth of pathogen is decreased by increasing or decreasing the pathogen from the pH 6. Here, the excellent sporulation was recorded at pH 6, and there was no sporulation below the pH 5 and above pH 9. And this 4 graph shows the fresh weight and dry weight of the pathogen at different media and different pH. The best media for the fresh weight and dry weight is PDA, and the best pH for the fresh weight, mycelial fresh weight and dry weight is pH 6. And this also is similar with the mycelial growth and sporulation of the pathogens. And this is a photograph showing the growth of the pathogen at different media and at different pH. While coming to the different temperature, among 6 different temperatures tested, like 15 to 40 degrees Celsius, it was seen that there was a quite large variation in the growth of the pathogen after 7 days. Temperatures, you can see in this graph clearly shows that temperatures from 20 degrees Celsius to 30 degrees Celsius was found to be the most favorable for the growth of the pathogen, while the highest growth of the pathogen was recorded at 30 degrees Celsius with highest sporulation. And finally, after characterization of my pathogen, after studying the characteristics of my pathogen, I have gone for the management part. In this, in vitro efficacy of various agrochemicals on the growth of the Fusarium modem was conducted by using food poisoning technique. I have taken fungicides, insecticides, herbicides, and combination of herbicides. Fungicides. In this, among the fungicides, I have taken copper oxychloride, mancozeb, and difenyl at different concentrations, and also combination of fungicides like azoxystrobin, difenoconazole, and propiconazole, difenoconazole, and metaloxyl chlorothalonil combination at each at 90 ppm, 180 ppm, and 360 ppm. And also herbicides and insecticides may have non-target effects on the soil-borne pathogens. So I have included insecticides and herbicides in my study. I have taken imidacloprid, fipronil, and chlorpyrifos and insecticides in this study, and also herbicides like glyphosate, paraquat, and pendimethalin were taken. In this graph, you can see that first graph shows that thiophanate methyl, among the fungicides used, thiophanate methyl has shows the maximum percentage of inhibition, growth inhibition of the pathogen at very low concentrations used. Among the 3 fungicides used, thiophanate methyl is used at very less concentration, but it shows the highest inhibition. And among the fungicide combinations, propiconazole and difenoconazole combinations is more effective than the other 2 combinations. But all the 3 combinations shows some effect on the growth of the pathogen. These chemicals move translaminarly and systematically in vascular system of plants so that it reduces the growth of the pathogen, and it also interferes with the electron transfer at quinol oxidation site so that it reduces the energy production and respiration of the pathogen. And this is a photograph showing in vitro efficacy of fungicides and fungicide combination on the growth of the Fusarium odum. While coming to the insecticides and herbicides, 3 insecticides and 3 herbicides were taken for the study at 3 different concentrations each. Among these, in insecticides, chlorpyrifos, and in herbicides, pendimethalin shows the highest percentage of inhibition. The inhibition may be due to the blocking of EPSPS enzyme in sickle acid pathway, that which ultimately affects the amino acid synthesis. This is the photograph showing the in vitro efficacy of insecticides and herbicides on the growth of the Fusarium odum pathogen. And while coming, I have also used the botanicals like plant extracts like garlic, ginger, and turmeric. Among these three, garlic has the highest percentage of inhibition, followed by the turmeric. This may be due to the presence of diallyl trisulfide and diallyl trisulfide in garlic, and also curcumin in turmeric. And while coming to the bioagents, I have taken the bioagents because Trichoderma species produces number of antibiotics like trichodermine, trichodermol, hersianamine, and also some cell wall degrading enzymes and some secondary metabolites. That secondary metabolites may trigger the activities like mycoparasitism, competition, rapid coagulation, etc. So that it has some effect on the growth of the pathogen. Among the 6 local isolates I have taken, Trichoderma hersianum given at the last shows the highest percentage of inhibition. Apart from the in vitro studies, I have also conducted the trial of field conditions using 2 best potent trichoderma isolates, that is Trichoderma hersianum and Trichoderma aspergillum, at 6 different treatments like soil treatment, seed treatment, and the combinations of both. Among this, all 6 plots treated with Trichoderma hersianum and Trichoderma aspergillum shows highest percentage of disease inhibition. Not only on disease inhibition, it also shows good results on germination percentage, plant height, and this trichoderma has effect on chlorophyll content also. While coming to the number of branches, trichoderma has no effect on the branches, and number of leaves, leaf length, and leaf breadth are also measured for calculating the leaf area index. In this, at 30th day, leaf area index is highest at plots treated with Trichoderma hersianum and Trichoderma aspergillum, but at the 60th day, leaf area index is more in the soil treated with Trichoderma hersianum. And finally, I conclude my presentation by saying that as this research includes the chemical characterization, the secondary metabolites identified here has more antifungal and antibacterial activities that not only that can be used for the crop protection purposes, and some other compounds present here can be quantified and can be utilized for the plant growth promoting products. So this pathogen can be used in a beneficial way also. And I have also identified the pathogen morphologically and molecularly, and um, and found that optimum pH is 6 and temperature is 30 degrees Celsius. And the findings also revealed that the application of herbicides and insecticides also have some non-target positive impact on the management of the pathogen Physarum udum. And the biocontrol agents, and it also shows the possibility of utilizing the native Trichoderma species for the management of the pigeon pea wilt, and also has some growth stimulating effects, so that it can be used for the sustainable plant disease management.
Thank you.
Finally, thank you, sir.
Now, Pankaj,
sir,
and Madam Srivastava, let us select two from this group to nominate the top two. From your side also, from my side and Madam's side, as soon as possible.
Sir, I'll just send the results through WhatsApp to you, sir. Will it
Yeah,
be?
that will do.
Okay, okay.
I will also send.
Yeah. Madam, my mobile number is different.
Okay, just let me know your mobile number then.
My mobile number is 700
700
254
54.
9989.
Okay, I'll send to this number.
Yeah.
I'll WhatsApp to this number.
Okay. So now we can go for next one other items for travel gun. We can finish it up also. Only one.
One, yeah, that can be done.
Sir, I just had one request.
Yes.
Sir, actually, there is only one presentation in APS Travel Grant. Na?
Yeah.
So maybe we can directly nominate him for this award.
Yeah.
Okay, that's no harm.
Let him present also. That will be better.
Would you not like to hear his presentation?
I think he should be given chance.
रिहर्सल हो जाएगा उनके लिए.
Okay.
Right. Yes, sir. He should be given a chance to present.
Yeah. Pankaj, invite him for. I think travel grant presentation also will continue at that time. Only one candidate is there.
Yes. So with the permission of chair, may I now invite Mr. R. Gautam Kumar.
Hello, sir.
Yes.
You can start your presentation.
Good afternoon, sir. Sir, am I audible?
Yes, very much.
Yes. Sir, are my slides visible, sir?
No, you have not shared. Oh, now it's coming.
Yes. Shall I start, sir?
Yeah.
Good afternoon, respected dignitaries present. Myself, R. Gautam Kumar, PhD student from the Assam Agriculture University, would like to present on the topic post-translational gene silencing using code protein gene-specific dsRNA molecules against coconut cucumber mosaic virus infecting Burjulokia crop of Assam. So the main background of this research is to stop the replication of the virus, where cucumber mosaic virus is a major virus infecting the Burjulokia crop. So these are the contents of my research. Coming to the rationale, every year the viruses cause huge economic losses, and also the plant viruses are very diverse and also use variety of strategies like the mode of replication and the mode of transmission made it very difficult to control the viruses. And also the viruses have been vectored by a great number of invertebrates involving some vectors, and also agricultural practices chemical control is also a harm to the crop. So there is a public demand for ecologically control of the viruses in a non-transgenic manner. So coming to the introduction, So Burjolokia is the major crop which is economically important to the state of Assam. Is recorded as the third hottest chili in the world. So the cucumber mosaic virus is considered as one of the most primitive viruses infecting the Burjolokia crop. So the virus is well known for its devastating nature and is even caused the yield loss of greater than 60% in the Burjolokia. So the CMV comes under the genus Cucumber virus of the family Bromaviridae and is mainly transmitted by the aphids in a non-persistent manner. So cucumber mosaic virus is a single-strand RNA positive strand virus which is non-enveloped and is spherical in shape, about 29 nanometer diameter, and is also cucumber mosaic viruses is divided into three segments, that is RNA1 and RNA2 and RNA3, which is having a 3'-tRNA-like structure and a 5' cap. So these are the gene functions of the cucumber mosaic virus, whereas the RNA1 and RNA2 encodes for the proteins 1A and 2B, which is mainly involved in genome replication and internal transcription, whereas the ORF2B is a viral suppressor gene for RNA silencing, whereas the RNA3 and RNA4 are translated into movement protein and capsid proteins. So in our research, we mainly targeted the code protein gene of the cucumber mosaic virus, as it mainly involves in the RNA encapsulation and cell-to-cell movement. So when we develop a double-strand RNA corresponding to the code protein gene, we can able to regulate the uncoating of the virus and where we can reduce the spread of the virus in the plant. So this taking this as the background. So this is a report of CM in Burjolokia, Northeast India, which was reported by Prashant, Bora, and Gautam. And these are the symptoms where we collected the same from the field, which are showing the symptoms like reduced plant growth, filiform leaves, and mosaic pattern of leaves. Coming to the background of the research, the RNA initially it was called as co-suppression by Jorgensen and laterally named as RNA interference or post-transcription gene silencing was discovered by Andrew Fire and Craig Mello in Cinerabditis elegans. RNA is a sequence-specific process which specifically targets the mRNA degradation. So it is initially induced by the double-strand RNA molecule. When a double-strand RNA molecule is introduced into the cell, it will be cleaved by dicer into a small molecule that is known as siRNA. So these siRNA molecules move into a complex known as RISC complex, which is known as RNA inducing silencing complex, where the double-stranded siRNA cleaves into passenger strand and the guide strand. So these guide strand will move to the translation machinery, which is occurring in the cytoplasm. So whenever the passenger strand and the mRNA, which is transcribing, binds, there is the enzyme ribonuclease H, which cleaves the translation pathway, thereby degrading the protein. So the protein is not translated in the further. So this is the background of our research. So taking this is our research means a long dsRNA molecule we will synthesize artificially and will introduce into the cell, and then the process will take place. So coming to the objectives of the work, development of a double-strand RNA construct for CMV resistance in Burjolokia. And the second objective is, after constructing a dsRNA, a proof of concept studies for CMV silencing in Burjolokia using dsRNA as a strategy. So for the development of the double-stranded RNA, so we have isolated the total RNA from the plant. So we have used two methods. One is trisal method and one is kit method. So in both the methods, we have isolated an abundant amount of RNA. So after obtaining of the RNA, we have estimated by quantitative analysis and qualitative analysis. So this is the gel picture of the total RNA. So we can see the abundant amount of RNA is present in the plant as well as the virus. So we want to amplify the CME virus. So we have synthesized the specific is it the code protein gene. So upon amplification, this CP region will show a 593 amplicon product. So this will be used as a target. So after synthesizing the primer, we have kept the efficiency of the primer in a software that is known as siRNA software. So it is showing when it produces amplicons. So these are the number of possible siRNAs will be present in our sequence. So with this picture, we are confirming that an ample number of siRNAs will be produced from our primer sequence. So after synthesizing of RNA, we have made it to DNA, that is by reverse transcription PCR. So from the transcription of PCR, we have again conducted primers. So after conducting of the PCR, we have loaded the samples in the gel electrophoresis. And after we get the bands, we have eluted with the scalpel, sterile scalpel. So this is the picture showing the amplification of the code protein gene, which is 593 base pair region. So this code protein gene is specific for the CMV. So after confirming of the code protein gene, we have confirmed the code protein gene into, cloned the code protein gene into a vector that is known as PT7 vector. This vector is having a restriction slice like ECOR1 and BAMH1. So taking this, for the process of cloning, the first step is ligation. So our PCR product is ligated into the vector PTZ. So this is the protocol followed for the ligation. So after the ligation, we have introduced into the DH5 alpha cells. So these are the preparation of competent cells, DH5 alpha strain. So after preparation of DH5 alpha strain, we have transformed our ligated product into the bacterial cell. So this is the transformation protocol. So after transformation, we have spread the bacterial cell on a LB agar plate. So on the next day at 37 degrees overnight culture, so we can see the colonies harboring our plasmid cell. So to confirm whether our gene of interest is present in the vector or not, we will extract the plasmids from the colonies. So this is the plasmid isolation from the colonies. After plasmid isolation, to know the gene of interest is present in the vector or not, we will go for the restriction digestion analysis. So restriction digestion analysis is done by two enzymes, that is BAMH1 and ECOR1. So these are the protocol followed for the restriction digestion analysis. So this is the result obtained from the restriction digestion analysis. Here we can see the gene is the 2.9 kb, that is PTZ vector, and this is a gene product. Here we can confirm that our gene of interest is present in the vector. So after confirmation of the restriction digestion analysis, we have done sequencing. So this is the protocol followed for the sequencing. So after in sequencing, we have added a product that is known as big dye terminator. So after following the procedure, we have loaded in this genetic analyzer. So we have obtained the results. So after getting the results, we have done blast in the NCBI website. So here it is confirming the product is having cucumber mosaic virus. After confirming, we again subcloned into the L4440 vector. Before we have cloned to confirm whether our gene of interest is cloning or not, whether the restriction site is at the perfect position or not, we have confirmed. So after confirm, we have again subcloned into L4440 vector. This vector and in this vector, the ligation and the transformation was followed as per previous. And also to confirm whether the gene of interest is present in L3440 vector, we have constructed the restriction digestion analysis. So in this restriction digestion analysis, we have done two restriction enzymes, that is BGL2 and XHO1. So this is the picture showing the restriction digestion analysis. This is the vector of L4440 and this is our gene of interest. So why we have introduced our gene of interest into L4440 vector is, so L4440 vector is having a specific property, that is having a T7 promoters at the adjacent to the gene of interest. So we can see the T7 promoters at the adjacent to our PCR product. So this is like an inducible operand concept. When we induce this transcription box with IPTZ, this T7 promoter molecules will move in opposite direction. This one this direction and this one this direction. So this is our DNA, which is our PCR product. So this is our DNA, which is a PCR product. And upon induction with IPTZ, these T7 promoters will operate, and this DNA will be converted into double-stranded RNA in the both directions. So this is our dsRNA molecules. So this is the protocol followed for the production of dsRNA. So after the production of dsRNA, we are pelleting out the dsRNA molecules. After pelleting out, we have added the nucleus-free water so that we have got the dsRNA. So this is the gel picture showing the dsRNA in the gel electrophoresis. So we can see the molecule which is induction with IPTG, and this is the molecule which we not kept the IPTG. So after confirming the dsRNA, we have gone for the field in the greenhouse condition test on the Burjolokia plants. So these are the plants of Burjolokia were grown in the net house to proceed for the dsRNA inoculation experiment. So seedlings were grown under greenhouse conditions and the plants were observed daily, and the seedlings at the age of 20 to 30 days were subject to dsRNA inoculation. So the experiment was conducted into three treatments. That is treatment 1, which is only the virus inoculated, that is CMV, which is named as non-vaccinated plants. And that is the treatment 2. Along with CMV, we also introduced the dsRNA, which was synthesized. And in the treatment 3, there is no CMV and no dsRNA, only the water we have given. So DSR application strategy. So to apply the dsRNA, we have first mechanically inoculated by rubbing with silite powder. We are make an injury. After making injury, we have applied the dsRNA onto the plant leaves. So this is the photograph taken at the 30 days. So here it is showing the plants which are inoculated only with the virus as showing the typical virus symptoms, whereas the plants which is inoculated with the virus and dsRNA showing the healthy growth, which indicates that the virus is suppressed by the dsRNA molecules, which indicates that the dsRNA is working efficiently. So these are the readings taken at the 30 dpa, which show that CMV inoculated plants have shown the reduced leaf size, whereas the CMV with the dsRNA inoculated cells show the more leaf area compared to CMV. And this is the plant height, showing that the decreased plant height is observed in CMV plants, whereas more height is observed in CMV plus dsRNA plants. So whether to check whether in a molecular level, whether the virus is present in the both the plants, we have done one-step RT-PCR. That means the leaves from the vaccinated plants and the non-vaccinated plants were collected, and again PCR was done. So after the PCR, this is the result. Whereas in CMV inoculated plants, where the virus is present, is showing the bands indicating that the virus is present. Whereas in CMV DSR inoculated, there is no virus is present, indicating that the virus was suppressed. Coming to the discussion, whereas according to Tenlode et al., the direct application of in vitro produced dsRNA derived from the virus sequence under plant tissue at the time of virus inoculation confers resistance against homologous virus. So, according to Kaldis et al., a tobacco mosaic virus was suppressed by non-transgenic approach by using double-strand RNA molecules. The results are in conformity with the findings of Konkalla et al., where the external application of dsRNA molecules from tobacco mosaic virus, that is from two genes P126 and CP genes, confers significant resistance against TME infection. So these are the conclusions. CMV core protein gene could be successfully detected from the Burjulaga fields, and the sequencing results was confirmed from the isolated CMV sequence of the Jorhat, and the 90% similarity was obtained. The CP gene was successfully cloned into L3440 vector, and the subcloning was confirmed by the restriction digestion analysis using BGL to XH1 restriction engines. Conclusion: the double-strand RNA was successfully produced by E. coli using T-cell RNA polymerase produced by Allodakis. When dsRNA exogenously applied on the leaves of Burjulaga along with the virus replication has reduced. The vaccination strategy could further be tested against different plant viruses impacting economically important crops where virus occurs and cause considerable losses. The RNA-based vaccination approach reported here could enhance the effectiveness of plant protection schemes for chili plants in nurseries and fields against CMV. So in future, these dsRNA vaccines could be modified as a dsRNA pesticides, where they could be applied in a field level like a pesticide strategy, so it would be effective against the plant viruses. So with this, I would like to conclude. Thank you.
Thank you. Finally, we came to an end of all these presentations and competitions. From my side also, I offer my heartiest thanks to Ao and Susanto for your kind help. And Madam Srivastava and Pankaj, thank you to you also for your participation and helping in conducting the competition. Now I'll expect Pankaj and Madam Srivastava will send their results. I will compile and I'll forward it to Susanta and Ao. Is it okay?
Okay. Sir, I have already sent the results. So you can check your WhatsApp.
I have also sent to you, sir, WhatsApp to you. Yeah. And I also express my sincere thanks to the organizer and to you for giving me this opportunity and associate with this special occasion. So thank you very much to all.
Thank you. Thank
I
you.
have another meeting, so I am leaving, sir, because I am in a hurry. Yeah. So I have another meeting. Yeah. I am leaving now. So thank you very much.
Okay. Okay, madam.
Thank you, sir,
actually, we took a lot of your time.
Thank you, sir, and a big thank you to Tia sir and Dr. Sushant for giving me this opportunity. Thanks a lot.
Yeah. In fact, we are thankful to you, and we are not getting time and opportunity to thank you. In fact, you have said for a very long time, and there are many technical issues also, and patiently and sincerely evaluating the presenters. It has been a tough job, in fact. So we are very thankful to you, all of you.
Thank you. Thank
you. And you have given us the opportunity. So from my side also, I offer my thanks to all of you, all the organizers.
Yes, sir. Yes, sir. Thank you. Thank you. Thank you to all three of you for responding positively and offering your moral support plus your physical presence here of the virtual symposium. Thank you so much.
I'll WhatsApp you.
Okay, yes, sir. Yes, sir.
I'll send the final results through WhatsApp.
Yes, sir. Yes, sir. We will do that. Yes, sir. So we'll wait for the result to start the validatory function.
Today? At what time?
Yes. Now we can also start immediately if you want.
Give little break. I'll see the results whether I received or not. Then I'll coordinate. They have sent. I'll compile and then
Yes, when you compile, we can go ahead with the program then.
Acha, TR is having a WhatsApp?
Yes, sir. Yes, sir. I have.
Or Susan.
But we will give time, sir. We will give time to you to announce during the valedictory program.
You can also announce the
You can
results.
announce it, no problem.
Okay.
With some remarks.
No, from IPS secretary side they will announce, or from your side you announce?
From Jonal chairperson, Jonal president and councillor will be announcing.
Yeah, they should announce.
Yes.
We are judging only. Just we will pass on the result. They will announce.
Yeah.
Okay, sir.
Okay, then. Okay, that is also okay. Then, Secretary, sir, shall we just go ahead with the valedictory program?
So then when this at least 15 minutes pause you can give without disconnecting, because the judge will take some time for compilation of this result.
Yeah.
If he has to declare, because you have to declare these results in this valedictory only.
It can be started, sir. No problem.
So,
Yes, sir. Meantime, sir, we have the result of the oral presentation. The
Yes, yeah, exactly.
Yeah, we can continue if Dr. Bhagavati can provide you that result of this.
Later on.
Yeah. At last also we can declare.
Yes, sir. Yes, sir.
Okay, okay.
And sir, we have a very senior CPS member, a retired university, with us for the valedictory.
Oh, he's very good.
So he will also be here as the guest of honor, and he will also address our valedictory program.
It's good, very good.
So we will just quickly get down the results that is sent by the committee members, and we will just proceed with the valedictory program if you agree, sir.
Yes, it will be better to save the time. We can continue. You go ahead.
Yes, sir. Yes, sir. Yes, sir. Our Singh sir has not been able to join us yet. I'll just contact him.
Fifteen, 15, 30, 30, 40, 46. Sixteen, 14 is 30, 44. 40, 30, 40, 44. Next again.
Yes, sir. Good afternoon, sir. Thanks for joining.
Okay.
Yes, sir. So we are just about to start our valedictory program. We are just collecting the results of the different competitions and award competitions. And shortly, in a while from now, we'll starting.
Okay.
So thank you so much once again for joining.
Okay.
Sir, you can, Viravanta sir, good afternoon.
Hello, good afternoon.
This is Robin Gogoi from Delhi.
Oh, sure. Because I got a news after calling by the Professor T. How. I did not know about that.
Okay, yeah.
You can remember.
Yeah,
Actually, sir. Actually, sir, Dr. Robin Gogoi, sir, is the current Secretary of Indian Phytopathological Society.
I know, because I long back 2006 I was the zonal president for Indian Phytopathological Society. By that time, my friend Chatterjee was the secretary.
Yes. Right.
D. Agrawal and all,
Right.
The Agrawal, we were quite friendly those days. So I used to go to Pusa Institute, used to stay there with them and spend time. So I retired in 2015 from Manipur University. So after that, I don't have much connection. Anyhow, it is very good that you have you remember me. That is one good thing.
Yes, sir. We were missing you since yesterday, and so we had to call you up. We have sent all the informations in your official email provided by the IPS headquarter and the address database. According to that, we have sent you the message. But we didn't see you yesterday, so we missed you, and I had to call you. And thank you so much for spending your time.
Anyhow, because of you, I have to join in because I was not much aware that what is going on this about this function.
Yes,
Anyhow,
sir. Yes, sir. So now that you have joined, and our respected secretary, IPS,
Susan Banik, who is Susan Banik?
So shall we start the program now?
No problem. Go ahead.
Okay, sir. Thank you, sir. Then I'll invite our counselor, Northeast Zone IPS, Dr. Sushanta Banik, to
glad to see you, Banik.
Yes, sir. So a very good afternoon to all, and we welcome everyone, Professor Viravanta Singh and Dr. Robin Gogoi, Secretary IPS, and all the senior professors who have joined us, the students, and other delegates. So for last 2 days, we have been holding this virtual symposium. We had inaugural function yesterday at 10 o'clock, and it was immediately followed by technical keynotes lecture session. We had two very eminent speakers who talked about microbial consortium and role of microbes in plant disease management, Professor L.C. Bora and Professor D.K. Jha. Then it was followed by a lunch break, and we had technical session starting from 1:30. There we had one lead lecture given by Dr. Sushil Kumar Sharma, scientist, ICR Research Complex, NEH, Manipur Center. And there are two oral presenters in the technical session 1. The session was chaired by Professor K.N. Bhagavati and co-chaired by Dr. T. Rajesh. Then we had two technical sessions combined, plant pathogen interaction and innovations and future technologies in plant disease management. Because we did not receive any abstract in these two sessions, we had to combine them, and this combined session was chaired by Professor R.K. Tomisana Devi and co-chaired by Dr. P. Raja, Associate Professor, College of Forestry in Pasigat. Then we had two lead lectures in that session. One lead lecture was given by Dr. Tasvina Rahman Bora, and the second one was given by Dr. Pranab Datta. The third technical session was on land disease management in organic agriculture. The session was chaired by Professor L. Daiho, Department of Plant Pathology, Sasar, Nagaland University, and co-chaired by Dr. Narula Pongener, Associate Professor in the department. And there was a lead lecture delivered by Dr. Pankaj Baiswar, Senior Scientist, ICR Research Complex, Northeastern Hill Region, Umiam, Meghalaya. And there were 5 oral presenters by various delegates. Now, that was yesterday, and today we started the day with e-poster session. There were altogether 11 presenters who presented their e-posters, and this e-poster session was evaluated by a committee, e-poster evaluation committee convened by Dr. Nongdren Khomba Singh Laikangbam, Associate Professor and Head, Department of Plant Pathology, CAU. And the members of the committee were Dr. Pranab Datta and Dr. Lanje Wangdi. They evaluated, and we have got the results of the e-poster presentations. Then it was immediately followed by the award competition, Professor M.J. Narasimhan Academic Award Contest. We had 8 presentations altogether, and 1 APS travel sponsorship for PhD students. Only 1 presentation was there, which ended just a while ago. Now we have got the results of oral presentations and e-poster presentations. We are yet to receive the result of Professor M.J. Narasimhan Academic Award Contest. So before we declare the results, and the time we wait for the results of Professor M.J. Narasimhan Academic Award Contest, meantime we'll proceed with the valedictory function. And we are today really privileged that we got Professor N. Iravanta Singh in a very short notice, in fact, very short notice. And he is a retired professor in higher academic grade of Life Science, School of Life Science in Manipur University. He is a fellow of Indian Phytopathological Society. And in fact, there are many societies for which he was a fellow, and I'll name them. Then Indian College of Allergy, Asthma and Immunology, Indian Aerobiological Society, International Bioresource Society, the Linnean Society of London, the Indian Mycological Society, Society of Biology, then Chartered Biologist, Chartered Scientist, UK. So he has been there as a fellow for all these societies. And now, sir, may we invite you to give your address kindly for this valedictory function?
Well, thank you very much, Dr. Bhanik. I'm also really thankful to Dr. Professor T. Au, because until unless I received that phone, I was in the town with some work. So I received that. I thought this must be from Notice, this number. Luckily, he introduced himself. You know that Tian Jam, what is called one of my students who was there in the Pasigat, and Meji Fema, he was a acting dean, or he was dean also. He is now registrar in charge or in the Nagaland University. What is I forgot his name. He was my student in St. Edmund's College, Shillong. You know, I'm very happy that last 2 days you have been doing a lot of brainstorming work on the plant disease management experience and aspiration. Well, for the plant pathologists of the Northeast India, we need a lot of the cooperation. We know the interaction. Individually, we have done good work. I know many of our young people have done very good work, but we do not know what type of the work, what is going on in those laboratories in the entire knowledge. Well, this is one of the factors that our things are not represented properly in the national fora. Well, when few years back, when I was in the Manipur University, I used to go very often to the Pusa Institute and spend time and interact with the senior scientists and exchange our works with them. So, but unluckily, our young people from the knowledge and also some of the friends like the Ken Bhagavati was a very good friend of mine. When I was in his Manipur University, everywhere I used to call him and interact with him. I go to their university, the Assam Agricultural University, for PhD evaluation, education of the PhD work and so. So we had good friends like the B.K. Datta from Assam University. He is also fellow of the Indian Pathopathological Society. He's member of the NEC, I remember. Like, I saw some of the persons like, I was the student of Professor H.K. Barua. He was the Vice Chancellor of the Guwahati University in long back, that is 74 to 79, I think he was like that. So many of his students were doing very good work in the entire knowledge. They have been spreading, like in Tribhuvan University, Islam was doing very good work. Then Ikramuddin Ahmad in Nagaland, he used to do that very good work. He was a Nagaland Science College. Very good work. Those people, D.N. Bardaloy in Tokla Institute, he was doing. So basically, my training was in aerobiology with special reference to the plant pathology. That was the thing. I was the president of the Indian Aerobiological Society 2007 to 10. Various aspects of the plant diseases, especially the plant disease forecasting, and also the disease management, forecasting in advance, what type of the chemicals you have to use for the plants, these were predicted during my studies. Many of our students have done good work. Half a dozen of my students come from agricultural backgrounds. They were doing very good work and they have been placed in different organizations in the country also. So therefore, I feel in this forum, we need to have our contact address. This is very important. Though I'm a retired person, but still I'm interested. We should have the contact interaction among ourselves and see what are the lacuna lacking behind in doing the research in this part of the country. A good number of the research projects, as much as the 16 major research projects related to plant pathology I have been doing in my career in Manipur University. But in these times, so in those days, I had a lot of interactions with the persons from different part of the country, especially from the Maharashtra region, especially from the Tamil Nadu, like Coimbatore, from Coimbatore, then from Andhra University. These are the people we interact each other, and also whenever we go to defend our projects, many of the non-persons are in the task force, in the panel of those experts. So therefore, I was very lucky in that way because I could get through those research projects. Therefore, I appeal to all the younger generation of our area that we try to have contact with those established plant pathologists in the country and try to join with them and interact with them. Whatever you have any your lacuna, that will help you in preparing the good projects. You see, the funding agents, I tell you, the funding agents, they are especially interested in the impact. Whatever money they spend on us, that will be applied, that will be useful to the society in general and to the farmers in particular. Therefore, we should know what is the application of the project you are going to submit to the funding agency. When you have that concept very clearly, through that they will easily sanction your projects. This is one aspect I want to give this suggestion to the younger plant pathologists of these regions. This is one aspect, and also try to consult the established or the plant pathologists in the country in general and in this region, Northeast regions. That is very important because we will not be able to do all the aspects of the work in our lifetimes, but we know what are the lacunae lacking behind with us and what are the lacunae that we should work out. This is very important. Therefore, with this, some of the comments in my mind, I feel that the disease management is very, very important. We should have the control of the diseases now really is very difficult. We have to manage how to manage this plant disease. That is the main aspects of the coming times. Then we have the experience, and also there are some aspirations, with some clear aspiration we should manage. Therefore, the management of the plant disease is not so easy task. We have to face a lot of the ups and downs, and lots of we have to think over various aspects. So, for example, we have now the recent aspects, you see, not only the plant pathologist, we have to have the interaction with the plant physiologist. Even we have the latest, what is that aspect? Your agriculture? No, you can have it. No, she told you. And you see some aspects like technology application with the help of the software and hardware application. We need not go through, find out the phosphorus content, iron content, macro and macro elements. These are to be with the help of the softwares developed by the younger generations of the Northeast. You see, the software technology of the India, Guwahati, they are funding some of those projects up to 25 lakhs to the young people who did work with the agriculture related work with the help of their technology, especially the hardware and software combined together. They are able to forecast how much percentage of the water is required for this plot of land and how much the amount of the phosphorus is needed, or especially in the tea cultivation that has been done. And also the plant, what are the factors that influence the disease, of the spread of the disease. So these are some of the aspects we have to take up, not only the disease aspects, the management aspect is very, very important. What about the irrigation facilities? What are the water facilities? What are the development factors that influences our disease in the disease components, especially in this Northeast India? We have about the tea is one of the important the material where we can earn a lot of the foreign exchanges. Therefore, now the scientists of the knowledge they have, especially those who are in the software technology part of India, Guwahati, there is a office. They are through the Minister of the Electronics, Government of India. They are giving a lot of the fund. So that we have to see also. So to get a fund from different funding agency, we have to look out what are the related aspects of the plant disease management apart from our classical botanical aspects, some other newly developed technologies we have to apply for the management of this plant disease. So these are some of the few things which come up in mind. I am very much happy that this Northeastern Zone of the Indian Phytopathological Society, especially the secretary from the National Secretary, Robin Gogoi, is here in this. So for giving me a chance to interact with you and share with you some of the thoughts I have in my mind, because I am not able to work on that. So these are some aspects. With these few comments, I once again thank all the organizer of today's what is called a virtual symposium of the Nordic IPS Nordic Zone. With these few words, I once again thank you all. Thank you very much.
Thank you so much, sir, for squeezing out some time for us in a very short notice. And it is always great to listen to persons like you. We learn a lot of things, good things. We get benefited. So I think your address, your sharing of thought has been beneficial to all the participants who are listening. So thank you so much, sir.
Okay. I will give you my address to Professor T. Ao.
Sir, got it, sir. Yes, we will keep in touch, sir.
Okay, that's fine.
Now, may I request our secretary, Dr. Robin Gogoi, to give his observation and remark. Dr. Robin Gogoi, sir.
Yes. Again. So I repeat. So I thank Dr. Susanta Bonik and also Dr. Tia Merenau for holding this responsibility to organize this Northeastern Zonal chapter of Indian Phytopathological Society. Despite of all difficulties and all odds, so they are able to make this program a great success. Now, so these viewers can see both of them. So as a zonal president and zonal councillor, they are sitting not in their own this campus. They are conducting this entire program from another campus, as they have informed me. This is from Dimapur campus, not the Medzifema campus. That's why we are very much thankful. So from headquarters, and the enthusiasm and also sincerity shown by all the participants really very incredible. And all the programs nicely conducted. They could invite very good speakers. So from the delivery of both the speakers, lead speakers, definitely, or I hope all the participants, particularly the students, will be benefited much. And I'm very much happy to see on the screen. So from other zones, see Dr. Tia and Dr. Susanta here. From Southern Zone, Dr. Uma Maheshwari and Dr. Nala Tambi. So they are from Southern Zone. They have already conducted their this symposium on the 1st and 2nd December. And also I have seen Dr. Sesa Kiran. He is representing from Central Zone of Indian Phytophthora Society. So he is now his zonal councillor, and they are also going to hold their turn, their function, the program in the month of January, in the new year, first week, that is 6th and 7th of January. So today only we have finalized their program and gave them the link. So Dr. Sesa, you will get the, I hope you have received our this mail reply with
Yes,
the
sir.
link.
I have already posted. I have already posted in pathology groups.
You are
You are so quick, Sesa.
Thank you.
And,
Thank you, sir.
so this is a very good opportunity to meet each other, even though it is not physical but virtual. But we are feeling that we are very close. I am just sitting 1 meter distance from all of you. So this is very more closest actually opportunity we are getting. This is blessed with this COVID-19. So this is good opportunity we are availing now.
That's
so,
right.
Dr. Iravanto Singh sir, he is definitely a very good guide, guardian, so very nicely spoken in front of us. And also Dr. Tombi Sinam, madam also sitting. Then my students also, many students I could witness on the screen. Very nice. I congratulate all of all the students. Dr. Uma, Dr. Nalatambi from headquarters, we must thank because they also very meticulously conducted in a huge way. But the Southern chapter was, it is not less than the National Symposium. Very nice. And that way we have seen the program prepared by Dr. Sesa Kiran and General President Dr. Srinivas Ulu. So they are also planning the program in a nice way. Concurrent session also they are planning to hold. And here again, I want to make announcements. So from yesterday, we have started for the first time in our this Indian Phytophthora Society this annual election in a virtual mode, means online mode. So those who are very well versed with this system, so they can enjoy by casting their vote online. Of course, this entertainment may not last longer time. Within 2 minutes, you can finish this entire steps. Very quickly you can do. There is no any hassle of printing the ballot paper, posting, spending 5 rupees in the stamp. So no any expenditure will be required. Only you have to spend your valuable time. That is only for 2 to 5 minutes. So I beg your this time for sincerely casting the vote in the coming future. So we hope so this online voting will be a grand success. We are trying. And similarly, those who are aspiring for the application of different awards, as in our this inaugural speech, I mentioned that IPS is offering total 17 awards, different awards in different categories. So if you would like to apply, so that will also be this online mode. From 15th of December will be opening this link. So you can upload enter this biodata along with credentials, certificates for competing in the different awards. And for the students particularly, I'd like to address, so really you keep your pace with great enthusiasm. So you are sitting in the one corner of this country. India is a very big country, but you don't feel that you are away from this mainland. So this virtual mode or this internet, it has become made a very small global village now. The way we are interacting with a fraction of second, we can have our discourse. So that way, so you can avail all the facilities. You don't lose heart and you keep your effort always make yourself very regular, meticulous, timely you try to do. So definitely the success will come to you. And from Pasighat campus also I saw Raza. He's also product of IRI. So he is continuing his service. So I think he is completely fascinated to the northeast, even though he belongs from Tamil Nadu. So very Raja. But here I am surprised that all the participants, they are connected, but they don't like to show their faces. I'll be very happy if I can see all of your faces. Otherwise, I'll feel very aloof. And Dr. Pankaj Baiswar and Tazvina, they also sincerely hold once a few years back this our IPS zone meeting symposium. And Dr. Pranab Datta in association with Dr. Ashok Bhattacharya, they also nicely hold this national symposium in 2018 in Jorhat campus. So that way we hope this CAU Imphal also in some coming days, they will be holding the national symposium. So the way their faculty and also the students are experiencing all these exposures, so definitely they will also be capable of holding this type of a grant program. So again, I must thank Dr. Rupankar Bhagavati. He's also like my senior brother. So he has given his valuable time in this judgment of these competitions, Narasimhan Award, Academic Award competition, as well as IPS APS travel grant. So here, many more I would like to say, but I don't like to continue. But here, some of the participants are really, I hope they are eagerly waiting to listen their results. Poster competition, this poster presentation competition, oral presentation competitions. So I hope this Dr. Susanta Banik and also Tia Mere now will announce shortly all the results of all the competitions, and they will definitely keep continue and very good close relation with us till at least this up to the national symposium, which we are planning to hold virtually in the month of March. So we are not yet declaring. We are just under process about organizing this national symposium. From Jorhat campus also, I saw Dr. Elsie Bara yesterday, and today I saw, just now I have seen Bharat Chandranath. So really, I'm feeling that I'm going back to my home place. Anyway, the entire program is a grand success. So I congratulate both this organizer, especially Dr. Tia and Dr. Susanta and their team. So I thank all of you once again. So keep it up for your future course of assignments. Thank you.
Thank you. Thank you very much, sir. You have actually finished part of my vote of thanks. So thank you so much for encouraging us and giving us directions and guidance as always. Now we have results ready. May I request our President, Professor N. Temara now to declare the results of e-poster competition, oral presentation, and the award contest for Professor M. J. Narasimhan and APS travel grant. We have been authorized by the evaluation committees to declare the results. So on their behalf, we are declaring the results. So time is yours.
Thank you so very much, Dr. Sushanta Banik, the counselor of the Indian Pathopathological Society, Northeast Zone chapter. It is my privilege to declare the results of oral presentation and Professor M. J. Narasimhan Academic Award and APS Travel Grant Award. I'm sure it was not a very easy work to do for the juries, because the contestants, the presenters were all equally very good. But in everything, there is someone who has to be chosen. And so let me, without any further ado, let me just go straight to the results. The result of the oral presentation. The third prize for the oral presentation goes to Dr. Joyshree Mohanda. She is the Assistant Chief Training Organizer, KVK, Garo Hills, Meghalaya. The second one is Mr. Kavisumi. He is a scientist, AICRP, Pigeon P, Nagaland University. And the first is a Miss Lepa Tep, PhD scholar, CIU, Barabani. So should I read the results once again?
Yes.
The first in the oral presentation is Miss Lepa Tep, PhD scholar, CIU, Parapani. Second, Mr. Kavisumi, scientist, AICRP, Pigeon P, Nagaland University. Third, Dr. Joyshree Mohanda, Assistant Chief Training Organizer, KVK West, Garo Hills, Meghalaya.
Okay.
Now, let us come to the result of Professor M.J. Narasimhan Academic Award. The first prize goes to Miss Arti Kumari.
Yes. Wow.
Second, Kakumani Soikia. So first, Arti Kumari. Second, Kakumani Soikia. Then the APS travel grant. There was one contestant, and the jury has selected him to proceed forward for a grant. And he is R. Gautam Kumar. R. Gautam Kumar from Assam Agricultural University.
He should have to show his face.
Yeah, now they should actually switch on their videos.
Okay, congratulations.
Yes. So with that, I would like to thank the jury once again for their hard work and giving a very fair and very fair result. And so with that, I would like to congratulate all the winners of both the award competition. Thank you. And the time goes now. The counselor can take over.
I want to add here, dear participant contestants who have actually declared this winner of different competitions. Especially, I want to mention here for this M.J. Narasimhan Academic Award Contest. This is actually nomination from your own zone.
Yes, sir.
So for final contest, they have to make presentation in the national symposium. Okay.
Yes, sir.
And similarly for IPS travel grant, Gautam, so I congratulate you. So your name will be sent to IPS headquarters, and final declaration about the selection will be made by them, not IPS. APS will be giving the declaration of the final results, because we have got 8 zones. From per zone 2 nominations should come, and there to a total 18 names will be proposing by the 16 names will be sending to the APS headquarters, but from northeastern zone only one yourself. So on an average 10 to 12 names will be proposed, and final results will be declared by APS. And one condition is there. So since you are single, so your name has been nominated for final selection, but you have to make your presentation in the national symposium. Okay, second time also. without your the presentation, so finally your name will not be sent to APS. Okay. And
one thing is important thing is all the award winners for contesting the national symposium, all should be the member of IPS. All the for Narasimhan Award contest and this APS travel grant, all should be member of the Indian Patrol Society.
This actually I wanted to add along with your results. So I wish all the best, all the three nominated candidate, so that definitely you can make a best presentation and you can show very strong competition in the final contest in the month of March. Okay, all the best.
All the best.
Yeah, thank you, sir, for the substantiation.
Thank you.
Sure, yeah. Thank you, sir.
Now it is time to declare the result of e-poster competition. So in the e-poster session, which was there in the morning, there were 11 presenters, and the jury have taken 3 names. I'll declare the results now. The third position goes to R. Lalrolui. R. Lalrolui, Department of Plant Pathology, School of Agricultural Science and Rural Development, Nagaland University. She presented on in vitro compatibility of Trichoderma aspergillum with fungicides and its effect against important plant pathogen. And the second position goes to Miss Barsha Devi. Barsha Devi from Department of Botany, Loknayak Omio Kumar Das College, Dhekia Juli, Assam. And the first position goes to Miss Emikam Wamut Nongtu, Department of Plant Pathology, School of Agricultural Science and Rural Development, Nagaland University, Mejifema. So congratulations to all the winners. So this is all about the results of the poster, oral and award contest.
Lipadev. We have seen the Lipadev, I think, along with Tasvina.
Congratulations.
Unmute
yourself.
Yeah, Dr. Tasmina.
First is Imika, the first prize winner from e-poster.
Okay,
congratulations.
Okay, thank you, sir.
Prasadevi also congratulations.
Then Arti Kumari also.
Arti Kumari.
Now all have joined.
After getting joined actually video.
Varsha also.
Lipa here.
Yes, Lipadev. Lipadev is the first, the winner of the oral presentation. Yes.
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 Nice to see you. Nice to see you.
Sir Arti Kumari.
Yeah.
Congratulations.
Yeah.
Congratulations.
Yes. Arti Kumari.
Yes.
Okay. Before I go to formal vote of thanks, now if anyone wants to share their observation or comments, remarks.
चलो जल्दी जल्दी आओ
Don't worry.
Yes, Madam Tombishana.
Yes, yes, sir.
You want to say something?
No, congratulations for my student. She is getting the first position in the oral presentation, and I just want to thank for nice selection of this drink. Chicken.
I think student also got first position, Arti Kumari.
Yes,
Yes, sir,
Aarti
yes,
Kumari
sir.
from AU.
Thank you, sir, for your nice question, because
No,
I'm
it was
really
a
happy.
really tough job for us to select
Yes, I
first,
can
second,
understand,
third.
sir. Thank you, thank you. And from my side, I would like to congratulate the organizing committee as well as the IPS
Yeah.
headquarters members. We
Yeah.
actually took a lot of pain to organize this symposium virtually. This is not a matter of joy, because we are in the Northeast and here the
Yeah.
internet connectivity is very, very problematic. Still,
Same.
organizing committee has completed very successfully. I congratulate one and all. Thank you all.
Thank you.
So, Dr. Uma from Wellington, Tamil Nadu, would you like to speak something? Because they have successfully completed and they are taking relax now.
Oh, yes, congratulations for awardees. And you conducted this symposium, virtual symposium, very nicely, even though from northeastern place. So congratulations to all zonal counselor and secretary, and especially the persons who are coordinating from Delhi. You have to thank them.
Yes.
Okay, thank you.
Dr. Sesa.
Good afternoon, everyone. Nice to see the organizers of this Northeast Zone. He's my best friend. I actually, just a little information. I completed my bachelor's in Bapatla in 96 to 2000, and I went to Dharwad. Susanta Banik joined for his MSc at my Bapatla campus. So we used to meet regularly for the past 20 years. So it is nice to see him organizing as counselor. So in the same year, we are acting as a counselor. So I congratulate him for successfully organizing, and also all the winners of the journal symposium. And I am very glad that my student, he is contesting for this Gautam. He is our past horticulture university graduate. It's nice to see the flavors of horticulture university coming into Assam Agricultural University and competing for this APS travel grant. And I also see this Madam Tasvina Bora. She's when I was doing PhD, she came to IRI. So it's like I'm seeing all my friends through this virtual meeting. Thank you, one and all.
Yeah, Dr. Raja, we had a very short glimpse. Can you show your face?
Dr. P. Raja.
Yeah, sir, I'm here. I was listening the whole program yesterday and today also. I was minutely watching all these. It was very nice connecting through all the pathologies through this virtual mode. So it is good to organize. Sometimes we have a shortfall of timings and other difficulties like this COVID pandemic situation. It is good to meet virtually, and then we can have a good interaction. I really thank for IPS for taking this step to connect all the people. Thank you so much, sir. Robin Corey, sir, and President, and even this Northeast Zone President and Councillor for taking such a step to connect all this. So thank you once again. There were a lot of students presenting very nicely. I could see their deliberation and research work. It was very nice. And most of them are 90%. I could see that they have all focusing only this organic method of management. It is good that in Northeast we are using we are not advising these chemicals and others. So students also doing this good works like taking up this back control. It is really appreciable, sir. Thank you so much. And so in near future also such a Zoom connectivity through this, we can connect a lot of programs. Thank you, sir.
Ripa, yeah, she's under her guidance.
Yeah. No, yeah.
So, and from my side, so actually I'm taking the mic from Susanta. At last, I will request Dr. Nalatambi to speak few words, because we are expecting, so you are also going to hold very shortly this APS travel grant contest, since it is pending from your zone. Unmute.
Unmuted.
Could you hear me? It's nice to see.
Your voice louder.
Hello, could you hear me? Oh.
Yeah.
Faint, very faint. Okay, I'll give chart because I think there is some problem in the.
Okay. Now it is coming, sir. Now voice is coming.
Okay. Thank you, Dr. Robin. It's nice to see that at least the Northeast group organized this meeting very nicely. I understand the difficulties, particularly when we worked with Dr. Raja and other group in Pasikat. Even getting this kind of advanced facilities to conduct this kind of meetings really nice to see. And there were days when even perhaps Dr. Raja will remember the days when we were sitting together working even without light and dark, and we were roaming here and there. So this is nice. Really, I should congratulate the president and the councillor. I think I understand that how much the effort they might have put to organize this meeting. Once again, I congratulate, and let us hope that the other zones also will do the similar kind of meeting. And since being organized from the Northeast, I had a little bit concern since we worked a collaborative project with the Northeast region, particularly the Pasikat. That's why last 2 days I was trying to attend this meeting. It really was well organized, nicely coordinated with all the people. I thank all the young students. They were very eager, very nice, and the language, the standard of language, it's very quite impressive. I congratulate all the students. Perhaps the new IRI coming in the Northeast Assam, I think they will compete with other IRI, even New Delhi and other place. Once again, I congratulate. Nice to see Dr. Raja. It's a good to see you. And thank you, Dr. Robin, for giving this opportunity. Thank you.
So this is the turn of this Dr. Susan.
Okay, sir. Thank you. It is nice to see the comments and feedbacks and know their observations, how they feel. But really, we had some problem technologically. We are still holding this in our mobile because time to time we need to switch to laptop, then laptop to mobile, mobile to laptop. So there are some technical issues yesterday. I think there are some noise disturbances. I think someone was commenting. So sorry for that. Except that, I think we had only the issue of prolonging the session because the participants, the presenters could not present in time due to their poor network. Anyway, the session prolonged, but somehow it could be finished. Now, actually, without any help from the IPS society, it could have been really impossible to have this virtual symposium. It is our first experience to hold virtual symposium or virtual thing of any kind. We have had virtual classes, but this is all different. We need to cooperate, we need to coordinate with so many people. Now, this is our first experience. I hope from our next events, we will try to coordinate better. Now, we thank, first of all, we thank the IPS headquarters for helping us in all manners and guiding us to hold these virtual symposium, especially Dr. Robin Gogoi, Secretary and IPS and organizing chairman of this year's virtual symposium. I thank Dr. P.K. Chakraborty, who, in spite of his busy schedule, gave us time.
Just I want to supplement Dr. Susanta.
Yes.
Just now I have received one message from our President Dr. P.K. Chakraborty and also Dr. Kalyan Mandal, Joint Secretary. Both of them are engaged now in one International Rice Symposium meeting. So that's why they could not join in this program. So both of them have conveyed very warm congratulations to both of you. Thank you.
Thank you. Thank you. Thank you, sir. Thank you so much, sir. So we are really thankful to Sir, Professor Dr. P.K. Chakravarti for being there in the inaugural function and sharing his thoughts, very insightful thoughts with the members. With the technical session, we had many people who accepted our invitation to be chair and co-chair, rapporteur. So we thank all of them who chaired, co-chaired, and acted as rapporteurs in all the sessions. And also the lead speakers who accepted our invitation in short notice and gave their lecture notes and presented beautifully in the technical sessions. I also thank the members of the evaluation committees, especially the IPS evaluation committees and the M.J. Narasimhan Academic Award Evaluation Committee, which have been quite long, and they have been patiently and sincerely hearing or listening to the presentations and giving their marks. And I really thankful to all the members, conveners, and the members of all the evaluation committees, and also the oral presentation evaluation committee chaired by Professor L. Daiho. We are thankful to him for his valuable time. He very quietly and sincerely conducted the session. Now, so all the participants who have been there with us since yesterday, without their participation, it was not possible to hold the symposium. And I also thank all the participants for really being patient and remaining hooked to this virtual form of symposium. So I thank each and everyone who have directly or indirectly helped us in organizing this virtual symposium. So thank you very much.
Okay, thank you. Thank you all. So hope to meet you all in National Symposium.
Yes,
it will be in the March 11 to 30 March in 2021.
Okay, sir. Yeah. Well, we are looking forward to the National Symposium, sir.
Sure, sir. Sure.
All of you are welcome.
Yes, and all the very best.
Yeah, thank you. Thank you.
Thank you, sir.
Thank you.
I think they will.